Thank you for coming to the second meeting of the Early House Conference. There's been a lot of discussion on the line, the webinar, many questions have come in from many patients and other people throughout the world. We're going to now begin session 3. Marcus?
Well, good morning, everybody. I'm going to continue with the biomarker group, question 2. But before I come to the background to our question, which is what criteria should be used to identify the most promising biomarkers for ALS clinical trials, I would like to present 3 hypotheses and maybe the evidence for it. Now, the first hypothesis is that this biomarker discussion is a hype. If you enter in PubMed ALS and biomarker, and I did this 2 days ago, you end up with 1,083 publications. I don't think there are that many biomarkers around that would justify that many publications about this topic. The second hypothesis is that the discussion about biomarker is dangerous for friendships. I received an email on November 3, 2015. We are not on the same page at all. And this was Moderator. But in the meantime, I can reassure you, we are on the same page, and we are actually very good friends. And as Martin mentioned yesterday, it was a lot of fun to work on this group. The third hypothesis is that, bias is an issue. A quote from a recent review in Muscle and Nerve: additional research is needed to ascertain its long-term value as a bona fide biomarker of disease progression. Now, this refers to EIM, and of course, the author here is Surat Ruradkov. I got approval from him that I could show this here at this meeting. Now, coming to the background of the second question, I think we all agree that the utility of biomarker depends on the stage of therapeutic development, with perhaps the greatest potentials in the mid-phase, which are essentially phase 2 clinical trials. And the questions we have to ask: has the experimental therapeutic reached and engaged its biological target? Has it affected the intended biological mechanism? And third, has it impacted the neurodegenerative process? So based on this, for the utilization of biomarkers, a distinction should be made between generic biomarkers of motor neuron loss, for instance, neurofilaments, versus those biomarkers which are specific for a particular mechanism of action, for instance, inflammation. Another issue is quality control. It's clear that the success and sound conclusions of a clinical trial depend on the quality of its experimental design and the measurements which are employed within this trial. And several studies actually have shown that quality control programs are beneficial for longitudinal, multi-center, and epidemiological studies. And one example for such a rigorous quality control was the xaliprodane trial. One outcome measure was the forced vital capacity. All the evaluators had to attend a mandatory training followed by intra and inter-test, retest reliability study, the results of which are shown here in this graph. You see a very reliable measure, very narrow confidence intervals. And this is where I think the biomarkers should go. So, rationale 1. There is a multitude of biomarkers, but few, or candidates, but few, if any, are ready for use in clinical trials to aid decisions about the advancement of therapeutics through the drug development pipeline. Partially, this is due to the absence of defined quality criteria against which biomarkers may be evaluated for their readiness for clinical trials. So the recommendation following this rationale are: putative biomarkers must meet certain minimum methodological criteria. They must be quantifiable, they must be practically implementable in a multi-center fashion, standardized, for instance, SOPs should be enforced, and show a high within center / rater and between center rater reliability. Rationale 2. Clinical information available at the time of trial enrollment, for instance, whether it's bulbar onset or latency from symptom onset to diagnosis, may be useful in predicting future prognosis. But significant heterogeneity in the rate of disease progression and in survival time at the level of the individual patient persists. So the recommendation derived from this is that prognostic biomarkers must provide valuable insight into prognosis over and above that which can be derived from readily available clinical information. Rationale 3. We all know ALS presents a clinical syndrome with multiple causes and potential diverse underlying upstream biological mechanisms, but this also provides a rationale for clinical trials in subsets of patients' populations most likely to benefit from a particular experimental therapeutic. The recommendation following this rationale is that putative predictive biomarkers should reasonably predict a future response to therapy based on biological principles. For instance, the presence of a particular genetic mutation identifies patients who benefit from a gene therapy that targets the genetic mutation. By contrast, established predictive biomarkers, we don't have them, must reliably predict response to treatment. Rationale 4. Robust measures of disease progression may aid clinical trials by providing a benchmark against which the impact of an experimental therapeutic can be measured. Established clinical measures, we do have them, for instance ALS-FRS, vital capacity, serve as useful measures of disease progression. But, and this is a major issue, they typically require large numbers of patients to be followed over an extended period of time. The recommendation following this is that biomarkers of disease progression should permit quantification of the longitudinal course of disease with greater sensitivity, which means a greater magnitude of change with less variability, if possible, than readily measured clinical outcomes. And finally, rationale 5. Biomarkers reflecting disease progression represent one potential type of biomarker, of pharmacodynamic biomarker. Biomarker quantifiably different in ALS patients compared to the healthy population, even without change over time, may serve as potential pharmacodynamic biomarkers. What we had in mind here were the neurofilaments. Potential pharmacodynamic biomarkers can only be validated as such once an effective therapeutic has been identified. The recommendation following here is that putative biomarkers of disease progression or pharmacodynamic effect must rest upon a foundation of longitudinal data. That's it.
So we're open to discussion.
There are some formatting comments.
So could you go back to rationale recommendation 3? So most of these aren't really recommendations, or they're not actionable. I mean, I think I have the idea of what you're trying to get at, but you know, putative predictive biomarkers don't do anything. They are, right? So they're not. Who's going to, you know? This is sort of a conclusion. This is the properties that you want them to have. And so, you know, we work that, like, investigators should only use biomarkers that, you know, fulfill these criteria, rather than saying the ideal biomarkers should look like this, because we're trying to make recommendations. So anyway, all the recommendations in this section are like that. So what should be investigated?
Okay. So we changed the wording. Thank you.
Yeah, sure, but I'm liberal about that. If you want to do a must or a minimum, I can look at it. Doesn't matter.
I can't hear you. Sorry.
I really like the framework. I think it captured most of the elements that we have access to currently absent effective therapy. So I had only a very minor point. On one of your early pieces, it was looking at saying that we must establish inter-center reliability, which would require participants moving, transferring from site to site to site. Is that your intention?
This is exactly the idea. I mean,
Excellent.
if we don't know the variability between the centers, we can't say anything about the noise we are creating with our data.
No, excellent. Great.
Any other comments?
No.
No burning issues here. Well, Dr. Benefit. Are you on this stage?
We are. Yeah. Okay. Thank you.
Great. So thanks very much, Martin and Marcus, for dealing with those first two questions. So the third question that this group tried to address was, what is the current status of candidate biomarkers for ALS therapy development? And if there's been not a lot of discussion about the first two questions, I'm anticipating that there may be more discussion and dialogue about these. In terms of background, for and this really is one of the recommendations out of the first question. And Gary, I hope that's okay, that a recommendation out of sort of the first in a series of questions can then become the background for a subsequent question. But the idea here is that for biomarkers to be useful in aiding decision-making in clinical trials, certain minimum methodological or quality criteria must be met. And Martin's been through this. This is question 2, recommendation 1, quantifiable, practically implementable in a multi-center fashion, standardized, and highly reliable. And then once we get beyond that, broad application will also require consideration of some pragmatic issues. So the burden of patient participation, investigator time in administering the biomarker, cost, the complexity of administration, the need for specialized equipment, and then the need for training and certification. And so in an effort to try to gather some of these data to answer the question about which of the current, what's the current status of these biomarker candidates, we sent out this table to the broader group, asking people to provide information about the putative role, what do we know about reliability, what's the sensitivity to change, trying to sort of gather this information, Hiroshi, in an evidence-informed fashion to answer this question. So, this is what came back, and this is just sort of first version of this, and this document went through multiple iterations, and what we very quickly learned is that understandably the data is not uniform, or the data are not uniform, and so very difficult to sort of standardize and homogenize and put into a simple format that gives us sort of a sense of the state of where we're at. So we sort of went back and thought a little bit more about, well, what are the sources of variability if we just try to pluck off that question? And so broadly, there's intra-subject. For a biochemical assay, there's an intra-assay and inter-assay. Then there's an evaluator effect within evaluator, between evaluators, between laboratories or between sites. And on the MR front, within a scanner and between scanners, and try to sort of informally ask for our different measures, biological fluid, imaging, neurophysiology, what are sort of the dominant or more influential areas of variability. So how do we then quantify that variability? And my apologies to the statisticians in the room. I got some advice from Eric yesterday, but wanted to dumb this down as much as I can, because the data are presented either as a coefficient of variation in the literature or as an intraclass correlation coefficient. And so the COV is really just a standardized measure of the dispersion of data around the mean. It's the standard deviation divided by the mean. And for the simple-minded folks like me, the interpretation here is that a lower number is better. For the intraclass correlation coefficient, this is the proportion of total variability that's attributable to variation between units, for example, between participants. And again, the interpretation here, actually this is not true, I've got that wrong, a lower number here, sorry, for intraclass correlation, a higher number is indeed better, meaning that most of the variation is biological, not the variation that's attributable to the assay or some element of the measurement. So what does this look like? What's the data look like for the biological fluids? And here's the coefficient of variation as a percent. And what you can see, here's some of the leading candidates, or the candidates for which we have the most data, right? So intra-assay, inter-assay variability, and these are in percentages. Within assay, 2.3%, between assay, up to 20%. Don't know inter-assay for neurofilament in blood, but up to 38%. And so I won't read through all of the numbers, but just to sort of cast your eye upon these and look at how many question marks there are, how little we know. These X's here mean that there's some data available, but just can't be summarized in the format of a coefficient of variation. So a lot of variability and a lot of missing data. What does this look like for neurophysiological measures? Most of this data is presented as intraclass correlation coefficients. So here's what we know for intra and inter evaluator. So reasonably good. Inter-laboratory or inter-site, a lot of missing data, still substantive variation. Sorry, this footnote is messed up here, but some of these data don't come from ALS. They come from healthy controls. They come from people with Duchenne dystrophy. For imaging, the data's there's even a greater paucity of data. We managed to collect some information about inter-evaluator off of the same scan for cervical spinal cord area, but no data for diffusion tensor, spectroscopy, other potential imaging measures. And then for these other measures that we asked about, SOD1 levels in spinal fluid, neurophysiological index, phrenic motor nerve response, nerve excitability, threshold tracking, MRS, DTI, really very little data to be able to begin to populate these sorts of tables. So I think this leads us to our rationale and recommendation, and there's only one. So the rationale, I think, is that the majority of published biomarker candidates have not yet met these key minimum methodological or quality criteria, and the evidence is part of what I've tried to show you. And so few, if any, biomarkers have reached a threshold that justifies recommendation for inclusion in clinical trials, and this is the important piece, to aid decision making. I don't think we've gotten to that level. And so I think the recommendation is that biomarker candidates should be incorporated into clinical trials for the purposes of learning, so that we acquire better data. And that additional experimental data must be accumulated in order to support the use of candidate biomarkers in future clinical trials for decision making. And Gary, I realize those are in the passive tense, so we have to sort of turn those around. But wanted to draw a distinction between utilization or incorporation of biomarkers for us to learn and be able to make better decisions, but that for none of these are we yet at a point where we have sufficient data to be able to use them to actually make decisions. And so I'll stop there and invite questions and comments.
Open to discussion.
Jeff. I can't hear you, sorry.
Michael, okay, so that was really super. I really enjoyed that summary. The question I would have, could you, and you did a ton of work, so you're not going to have this, you may have this answer, but if you compared what you just did to an established biomarker and say, you know, heart disease or something, what would that look like?
I don't know. It's a great question. I've long sort of wanted to do that, but this was kind of enough of an undertaking.
Yeah, okay.
I don't know, but I think it might be valuable for us as a field to know that. I don't know if anybody else does, but I don't.
Very nice, Michael. I have the feeling that many of us are seeing these ways of evaluating biomarkers for the first time. What I think you're showing us is a great approach to a more rigorous analysis of how valuable biomarkers are. And for me, this is like biomarker 101. I would encourage you and your group to develop this material into a review of where are we in biomarkers, because this kind of approach and standardizing it and putting it out there for comment, as it were, would I think be very helpful.
Great, thanks. I agree, and I think the whole group probably feels that way and something that we should follow up on.
So, yeah, thank you. So first, let me echo, I thought the previous talk and this one were excellent. So can I ask you a question then concerning imaging and advanced imaging, and then, you know, taking the different imaging types like MRS, MRI, DTI, and combining them for evaluating a new biomarker? Do you have any concept of whether you think that's truly feasible based on cost? Because you did list cost, I think it was your third point in your list of what a biomarker would really need to be. And can you use the experience in the MS field, where they clearly have used MRI as a biomarker, to help us understand if this approach that many people are taking in the room is really a feasible one for a new biomarker?
It's a great question, and I don't want to put anybody else on the spot, but I may ask Sanjay for help in answering this question, if he doesn't mind, because he's, I think, thought about this a lot more, and he's got an active study at the moment. I think there's still a lot of hope, but I don't think we're there yet, and I do worry about the cost and feasibility. But Sanjay, could you say a word?
Yeah, sure. Thanks, Michael. So we have the Canadian ALS Neuroimaging Consortium. It's a group of 5 centers currently, expanding to 7 centers in Canada, where we're actually doing just this. So it's a prospective study going to collect about over 100 subjects, equal number of controls over the next few years. It's actively recruiting right now, but this is exactly what we're doing, looking at DTI, MR spectroscopy, volumetrics, so various parameters you've alluded to, looking at inter-MR scanner and inter-scanner variability, in addition to looking at hypotheses relevant to ALS. So are there longitudinal changes? Are these predictive? Are they prognostic?
Yeah. And so, Ava, I mean, I think to some extent it depends on, I guess, how much this can be done across multi-centers. If somebody were proposing, you know, a biomarker based on a 7T MRI scanner, I'd say, well, those, you know, there are not too many of those at many centers, and that may not be that practical. But if you can get all these data on sort of a, you know, 45-minute scan and a 3T scanner, and it's robust to whether it's a G or a Siemens, and then maybe there is value. But it's the work that sort of Sanjay and others are doing that I think answer that question for us.
Yeah, that is the first step, you know, making sure that the scanners are the same field strength, 3T, all Siemens, GE, etc. So it's quite an undertaking to make sure everything is standardized to protocol. But it is possible. We're up and running. It'll take a few years. We'll have this data.
Michael, thanks. I think that's great synthesis. I love having all that data together. The suggestion I have is to encourage you in your group to put forward a few more recommendations about the types of data that would be valuable for evaluating a biomarker. The pieces that you've talked about so far are sort of the 0th level. Can we reproduce the same measurement in the same person by different, you know, at different sites? And what we also need to know is what has been talked about. Is it prognostic? So what's the relationship between baseline levels of these measures and someone's future course? What's the relationship over time? Is there concordance in these measures? Does it seem to track some of the dynamics of progression that we think are relevant to the disease? ALS, FRSR, FVC. And lastly, to make it useful is in the process of shortening and making more efficient our clinical trials, we'd like to know whether over the course of watching a given biomarker for, say, 3 months, is that predictive of the person's course over a year? Could we shorten our time course of trials by having this much more sensitive measure of their underlying physiology relative to their overall clinical progression? So those are things that I think you could articulate and specifically identify what are the types of data that should be presented in papers that evaluate biomarkers.
I agree. Thank you for those comments. I mean, I think Marcus dealt with one of those, your last point, I think, Eric, that when we think about prognostic markers, we really have to be thinking about what prognostic value they add over and above what we can already tell based on clinical measures. Because if you're going to undertake some expensive, complicated assay and it only tells you what we already know, then I think it really doesn't add value. And so I think things have to be looked at and compared to what do they add over and above the best the best information we can gather sort of at the bedside to begin with.
Yeah. So the caution there is to think clearly about how are you going to decide what's better. So if your biomarker says that this person's fine and ALS FRS says they got sicker, which which was right.
I think longitudinal data will tell us what was right. So I think these studies have to be set up in a prospective longitudinal fashion.
Right.
So
And
I agree.
I suspect that survival will be our final barometer.
Yeah,
So
I agree.
yeah, that makes these very long studies.
It does.
So I'd like to expand on your comment, Bob, and from yesterday's Martin's presentation, this biomarkers, obviously biomarker is critically important. And so I'd like to, we have now, there is no discussion about Marcus's, after Marcus's presentation, so maybe we should spend a few minutes more this biomarkers group, not just for our guidelines, but expand something more to organize. For example, Eric's bio statistic comment and Bob's comment, how we can move this forward. This is a momentum to do multi-centers. Already maybe Mike is doing multi-center studies, but more organized fashion to develop not just the clinical trials, but longitudinally many different neurophysiological and neuroimaging and biochemical biomarker. How we can present, first maybe we can present papers and move forward to direct us how we move internationally. And so just I'd like to open discussion to how move this step forward. So just I'd like to expand the discussions.
Actually, my question is related to that. So if I can get mine in here, then the discussion to a large extent has been related to prognostics. I know that we've talked about trying to incorporate these in interventional studies. But Michael, my question to you is, do you foresee in this, if we are opening it up, the other speaker could comment, if we are opening this up to interventional studies, could there be value in compounding the data to validate these markers? Could there be value in bringing these into longer duration, single arm access trials? So what I mean is that we're talking so far in the prognostic sense of perturbing the data with natural history outcomes of the disease. So my question is, could we add to that by perturbing the data with apparent responses or relative outcomes in interventional drug studies, not just the ones that are actively controlled, but some of the studies that we talked about yesterday that might be much longer duration, single arm, expanded access trials?
My own view, and this is not a discussion we had as a group, and so I think others should comment too, is I think any prospective systematic collection of data is an opportunity to advance the biomarker field. And so I think any study that's doing something sort of systematized, that's sort of following outcomes, and that's collecting that kind of rigorous data is an opportunity to be doing this. And I think that's part of the recommendation about learning. So maybe that recommendation can be broadened, that it's not just, I mean, I guess we framed it in the context of clinical trials because it's a clinical trial, the guideline that's being developed. But I think that there's an opportunity to learn, and we need to do a lot of learning in any such prospective undertaking.
Great. Yeah, I would think that it would be very different. I mean, this independent variable of outcomes within an interventional study for the treatment arm is totally different than looking just at the natural outcomes as a prognostic for the patient.
I agree. I mean, I think one of the challenges, though, is that if we don't know, for example, the natural history of a biochemical marker, and we don't know what the variability in the noise is, and you then try to interpret that data in a single-arm study, I think it's uninterpretable. I think that's part of the problem. But I think it's still an opportunity to learn, but we have to set up experiments in such a way that at the end of the day, we've learned something, right? And then we advance.
There was a recent paper, I think the German group looking at the neurofilament, and they looked at it diagnostically and over time. There wasn't change over time. It's like a state versus a diagnosis, and those are some of the harder issues. in terms of that. Dr. Coffey?
Yeah.
All right. So it's related to that discussion. I'm curious to get your thoughts or anyone from your group. And maybe this is not the most popular view, but I think one of the challenges here is kind of what you were hitting on before, that there are kind of two separate issues here. There's the drug development field doing clinical trials, and we'd like to have biomarkers to be more efficient in the drug development, but there's the validating and improving biomarkers. And I think not just in ALS, in a lot of fields, we confound those two. We try to do the validation of biomarkers in clinical trials looking at drugs, and I don't think, as a methodologist, I think you're never going to get to where you want to get until we kind of fundamentally change our scientific approach and look at biomarker studies as completely separate studies and say, if we want to have better biomarkers, we need to design to both explore and validate biomarkers and have those studies that are learning about biomarkers outside of the drug development world, not sticking them in and trying to like get everything within one study, which is more costly, has different funding. But I don't see how you can validate biomarkers in a study where you have an intervention, because if the intervention works, the biomarker might be changing with the intervention, it might not. And as you mentioned before, I don't see how you can disentangle that. So it's almost a paradigm shift where we need to think of this as a separate field of studies that need to be done and funded and resourced. I'm curious on your thoughts.
So I agree, but I also think that there is an opportunity to learn from clinical trials, because the unfortunate reality is that every therapeutic has not been effective. And so there are placebo groups, and two, if the therapeutic is not effective, I think in a sense it becomes more natural history data. But having said that, I agree. I think this is important enough that it warrants independent formal study to get it right and to be able to flesh out and provide the sort of data that we need to know how do we use these biomarker data.
Yeah, I think the only thing I would caution, because I agree fundamentally with what you're saying, but if there's a trial that's not negative, right, just remember, like statistically, you can't assume that there's no effect, right? You can just assume you didn't see the effect that you wanted. So it's possible that there's still some small intervention effects that didn't reach your level of significance that are somehow underlying.
I hear you. Marcus?
Just one brief comment. We did not talk about, you used the term validation, but we were not talking about validation. The first step is standardization and reliability testing. And once we have done this, then we can move on to validation or to explore the biomarkers. But the critical issue here is, and this is what I wanted to get at, as long as we do not have standardized the various candidates across centers, and as long as we do not know their reliability in a multi-center setting, we don't need to talk about validation or use in a clinical trial.
So, Michael, as you know, we're doing exactly what Dr. Coffey asked for. Biogen's doing a longitudinal methodology study with no intervention, with head-to-head comparison of the various electrophysiologic techniques, et cetera. I think it's worth taking an inventory of those current efforts, including your CREATE study. And it's actually, I think it's worth formally doing it, just sticking a list of those things out. And Jeff Rothstein's 1,000-person thing. There are some efforts going on. I think the question to throw out is, does ALS need an ADNI? So those of you who know from the Alzheimer's field that the ADNI initiative has really revolutionized the entire field of Alzheimer's drug development and biomarkers. Now, that's obviously a more prevalent disease, et cetera, but there's lessons learned. They have cut their teeth on a lot of difficult things. There are people in the room that are involved with that. So I think it's worth taking an inventory, and like your study, others, and sort of putting on their list what are the, and then Lucy Brune has been, excuse me, very helpful to sort of coordinate all these activities and try to make the pieces all fit together. So I think it's worth actually just taking an inventory of current efforts. and maybe identifying gaps for future efforts.
I agree, and it makes me think, although I didn't see you, that you were sitting at breakfast with Lucy and I this morning, because we were discussing just this very issue of the importance of taking that inventory. And there has been a discussion for a while about trying to establish sort of a biomarker consortium across the field. And I hesitate to use the term ADNI, although, because that has neuroimaging in the title, and although that will make Sanjay and others happy, I think it needs to, I know what you mean, but...
Because as you know, ADNI has actually, imaging became a very minor part of it.
I know, it's just an unfortunate term, but I agree.
It's the branding. It's the branding, though.
So I agree, and I think there is agreement, and I know that Lucy and others have been in discussions about how we might do this, but I agree, I think we would benefit as a field.
Me and the South Africans are always on the same page, Michael.
Indeed, indeed.
Bob Bowser from Barron Neurologic. So many of the comments are things that actually are to multiple levels ongoing in the field, and there are large multi-center studies, a lot that's occurring in Europe, and now a few, and there's another paper coming out of validating some of the neurofilaments across international, including the US and Europe. So there's a lot of activity out there, and as Don was alluding, there is a lot of other longitudinal studies that have already been in existence for a number of years. And I think the inventory is a great approach to try to figure out how can we work both together, what has been done already, what's the state of the art at the moment. I think some of the important things that happened now a number of years ago was really just generating SOPs for collecting, processing, and storing of samples. That has been implemented across the field, and especially throughout the NEALS consortium. And so we are at least collecting samples appropriately. So I think even when Jess mentioned about expanded access programs and the potential utility of biomarker work there, I think at the first level, even if we don't have yet the truly validated biomarkers that we all hope and want to get, if we're at least implementing SOPs in these studies, even if they're open label, and collecting samples and storing them properly, then they're available for use maybe a year later or something. If something's validated, then they can be used. So I think there's a lot of information that can be gleaned from such studies if they're done appropriately. So again, there's a number of these ongoing longitudinal collection studies. I mean, we have one that's 6 sites that's got about 130 patients enrolled that started 3 to 4 years ago. So, and there's more ongoing. I know Michael has the CREATE that just started. So there's a lot of opportunity, and I think as a field we just need to work together in order to maximize all the current efforts to date, and then to best strategize how to take these forward so that we can ultimately have these markers available for our future clinical trials.
I agree. But one question I wanted to put to you while you're there, and something that I don't know that we've wrestled with as a field, and I don't know whether we need to. So, for example, I understand, although I don't know the field well, that, for example, CSF amyloid beta levels will vary depending on the time of day that the sample is collected.
Yeah.
And do we need to worry about that, for example, with neurofilament? Has that been looked at all? I'm not aware of that data.
No, it needs to be done. I mean, those are great questions. That type of work needs to be done. I mean, internally, we've tried to collect samples at around the same time, but patients come in when they can come in, and so it makes those types of quality control issues important to know. So, but you're really, if you have patients over a 24-hour period that basically have a spinal tap for other reasons, and you draw out fluid over time, that can help address those questions. And that was what was done in the Alzheimer's field to identify that problem. The other thing that I did want to state, and now I forget what I was going to say. That's another ADNI problem. What's that? Alzheimer's. Yeah, that's an ADNI problem.
But maybe this is part of the recommendations that Hiroshi is asking us to come up with. Is what are some of those sort of specific, actionable sort of things that we as a community need to do, and how do we prioritize? Because there are a lot of sources of variability, and how do we decide where do we want to focus our efforts?
Because you mentioned that. So the other thing that we can consider is you have all the inter-reliability, inter-site variations that can exist. So, and that's one model to do. The other model is to collect the samples all in the same way, but send that to a validated central lab. And so
Absolutely.
now you're avoiding some of those
I agree.
All those variabilities across different labs. And so that's another model that we can consider as a group, that if we have something that we think is really interesting, you send it to a central lab that has assays validated.
I agree.
So from tactics back to strategy, Dr. Turner, and then I'm going. You're just standing up. Okay.
Okay. I think the reproducibility or reflecting the pathology or other things you presented is very wonderful at present, particularly for diagnostic markers or natural history. But from the viewpoint of surrogacy, against the surrogate, how surrogate the marker against the response of the clinical response under the certain drug, something like that. Probably the relationship between biomarker to the surrogacy of the response of the clinical base will require some other approach to assess how surrogate the clinical response. Probably it's another approach will be necessary to do that. How do you think that?
So I think we pulled back from talking about surrogacy, but I think I think we haven't got there as a field, in the sense of I think we have to do some of the foundational work to understand how these biomarkers perform before we can get to surrogacy. And surrogacy, it turns out, is actually more controversial from an FDA perspective, as I understand it. When we're asking about using biomarkers in phase 2 to help make decisions, that's uncontroversial from an FDA perspective. If we're wanting to use a biomarker in a phase 3 to seek drug approval, that raises a far more controversial issue, and I think raises the bar tremendously in terms of what we would need to show. And I think actually that our challenge as a field, and what we really need is, I think we need the biomarkers in phase 2 to help make decisions about what to take to phase 3. I don't know that our real challenge is the need for a surrogate marker in phase 3. I don't know that that's clear. I'd be interested in other people's thoughts. That doesn't seem to me to be where we should really focus our attention. So I think surrogacy is important, valuable, nice to think about, but perhaps not our priority as a field. But what do others think?
Just a quick question. What is the most promising biomarker that allows earlier diagnosis and criteria right now? So I do think the biggest role of biomarker right now is to make diagnosis earlier. And is there any discussion on that in your group?
So I think the group perhaps disagreed, in the sense that I didn't think, we as a group didn't think that the diagnostic issue, at least with respect to clinical trial guideline, was the key issue. Because the challenge, as you know, is getting patients to see a neurologist or to have somebody think about ALS early in the course of disease. By the time people show up to see a neurologist who knows something about the disease, the diagnosis is fairly clear. And most of the studies that have looked at diagnostic questions and biomarkers have sort of been looking in that population in which the diagnosis is already clear. And so I think we're even more in infancy than we perhaps realize from a diagnostic perspective. I think this will become more relevant as we can get patients into clinic and clinicians thinking about the diagnostic possibilities earlier, when the clinical diagnosis may be less apparent. And then I think that clinical or that diagnostic question will become much more important. But as a group, we didn't really address this at all. We sort of set it aside.
The group should consider the FDA guidance on biological. You talked about what does it take to have a good biomarker. I think that came up. And I think our rationale should include that website. And going along with what other people have said, we should probably have forward-looking statements, because a lot of people feel that the FDA guidance on development of biomarkers is not where we want to be. And so when we move our language forward in terms of what you were talking about, I think we have to at least cite that website, and then in many ways changed our wording in response to that.
So,
The ideas that you came up with.
So 2 comments. Firstly, I think that we felt that the conceptual framework provided by the FDA in terms of types of biomarkers, diagnostic, prognostic, predictive, pharmacodynamic, is a useful framework. But we've added to that the concept of a disease progression marker. What we did not do is focus on the FDA's guidance of qualification for biomarkers, because I think many of us think that with apologies to the FDA, isn't where it needs to be and is probably going to evolve, and didn't want to tie our recommendations into that framework. So we've sort of acknowledged and agreed to a large extent with FDA language, but have tried to sort of propose ideas and concepts and recommendations that are independent of that qualification process. So I think that was the mindset, which I think is what you're getting at.
Well, actually, you should be talking because you summarized all this in a very nice paper a couple of years ago, and I think that really sums it up. But I want to come back to the question that you asked, why do we need a biomarker? I think, at least to me, it is one of the more important reasons is to use them exactly as you say in very early pharmacological trials, to see whether a drug touches the disease process at all in a short trial in a limited number of patients. I don't think we will ever need a biomarker in a good phase 3 trial, because clinicians, nor patients, nor authorities are interested to know whether a drug affects a biomarker in this disease.
Agree 100%.
We need to know whether it makes patients better or protects them against progression or whatever.
I agree 100%.
I was just going to, I entirely agree. And to go back to your point about diagnostics, I think actually it would be very interesting to ask people to ask each other over coffee. You know, what is the question that ALS neurologists are asking in clinic? And there are a very few small group of patients, we all know who they are, where often boiling it down, we're after a marker of occult upper motor neuron involvement. That's the missing gap in diagnostics. It's not actually the main issue, can we have something that makes a test for ALS? And our question in clinic is not, is this person a healthy volunteer or not? You know, yet we do all our comparisons with healthy people. So it's much more about collecting what the real mimics are, recognizing that's a very small group, and the bigger picture is something that changes in a trial in 3 months rather than waiting for survival at 18.
Thank you very much, and we have to move on to the next question. For question 1, what is the role of alternative design, etc.? And we have discussed some of enrichment yesterday. And please.
So my name is James Berry. I'm going to just sort of introduce our group. We have a group of 6 questions plus a review of one of the guidelines from the last recommendations, looking at trial design. We have 6 different presenters who are each going to come up, and what I would invite them to do is sort of come up sort of during the discussion of the question before. You can come up, maybe sit here in the front so that we can make that transition quick. The other thing I would just note is that the recommendations in the slides for our questions have evolved slightly since the handout. So the recommendations may be formatted slightly differently for each of our questions and recommendations than they are in the handout. That just simply reflects the sort of evolving thoughts about these things as this meeting has gone along to some extent. Some of it is just moving to an active voice, et cetera, et cetera. People will try to sort of highlight the challenges that we had as a group in addressing a lot of these questions, because I think those are the points that we need the most comment on before we go back to do the final draft. So Lauren Zinman is going to get started with the first question.
Thanks, James. Let's see here. Okay, so alternative designs. A bit of background. I'll skip right to the enormous funds and resources that are required to take a drug from phase 1 all to phase 3. And obviously the people in this room know that tens of millions of dollars are required to get a drug through that process. And we have an increased number of candidate drugs for a very rare disease. So faced with these difficulties with incredible expense and a rare number of drugs, a solution can be alternative designs, which can reduce costs and expedite the discovery process. And there are a number of alternative designs, and this is not a complete list. What we've done is highlighted the more important designs that can be helpful in ALS. The first thing to mention is adaptive designs. And while normally we go from phase 1, phase 2, phase 3, 3 separate protocols, 3 separate IRB submissions, 3 separate contracts if there's multiple sites, this can all be combined into one adaptive design where you combine safety, tolerability, PK, PD, and dose selection into a single study with predefined criteria that goes from stage to stage. This was done in the ceftriaxone study, where looking at different pharmacokinetics would determine what happened in the next stage of the study. But there are a number of different adaptive designs. There's an adaptive biomarker-directed trial, which can be modified based on the short-term results of a pharmacodynamic biomarker. For example, let's say you're interested in a certain serum or CSF growth factor, a protein. What you could do is look and see which of those subjects actually had target engagement and take those subjects to go on to the next stage of the trial. There's adaptive randomization trials, which you heard about last night, where basically randomization is redistributed based on an interim analysis. For example, if you have placebo and then high, medium, and low dose, let's say everybody in the high dose is having toxicity and poor tolerability, at the 50% point, you can switch to 3 arms instead of 4. There are enrichment designs, and really what the aim of what you're trying to do here is you're trying to include subjects most likely to be treatment responsive, and this is informed by a biomarker. And this is really critical in a heterogeneous disease like ALS, where most of us feel that patients are not going to respond uniformly to a given therapeutic. And this is really nicely being done in Canada right now in a study led by Lawrence Korngut, where we're looking at a compound called pimozide, which in preclinical studies showed that seemed to have an effect at the neuromuscular junction. So the way this study is being enriched is all patients are getting repetitive nerve stimulation, and those subjects that have a decrement are being included in the study. So in other words, the study is being enriched for those patients where we believe the pathology is at the neuromuscular junction to increase the likelihood that your compound will have an effect. Futility designs, basically what you're doing here is you're reversing the null hypothesis. And the reason that helps is it gives you a more liberalized alpha significant value. And this is really helpful when the focus is on dismissing promising therapeutics that have little chance for success in the early phase of development. And this is obviously very advantageous in ALS given the chance that the compound is successful in the first place is quite low. So this is something that can be employed as well. There are these selection designs we heard about last night from Dr. Vickers, a pick a winner design which tests multiple promising therapeutics in a single trial. Big advantage in ALS with a limited number of eligible subjects and a growing number of promising therapeutics. It's also an advantage because you can use historical controls. So all patients will actually get a promising therapeutic, and we know how advantageous that is in terms of recruiting patients. Again, I mentioned historical controls. Bob Miller brought this up yesterday. This is good this is really advantageous in the early phases of ALS because the disease is linear. Again, all subjects get the active compound, which is great for recruitment and retention. But you've got to be really cautious with your controls given the evolving field. You have to make sure that your historical control population is representative of those patients in the trial. For example, are they getting access to BiPAP? Are they getting access to feeding tubes? Are they being followed in a multidisciplinary clinic like the patients in the trial? What some studies in Parkinson's disease have done is they use historical controls, but they also have a tiny, tiny group of a placebo group within the study just to validate that the historical controls are actually representative of what's going on in real time in the study, and that's something we can do as well. It limits the number of patients who are actually randomized to the placebo group. And then there are a number of other designs, lead-in designs, and Darravone used a lead-in design to select patients in the earliest phase of the disease and found a benefit. A time to failure design where you cross over from placebo to active, and this is what was employed in the lithium study. The advantage here is that patients will be spend a limited amount of time on placebo, and you can recruit patients by saying, pitching that, if you hit a certain failure point, you'll be crossed over to active group. It's hard to cross over the other way to go from active to placebo in a neurodegenerative disease because a compound could have a longer lasting effect, but for symptomatic studies, you can use a crossover both ways. So our rationale is alternative study designs can more efficiently and cost-effectively evaluate promising therapeutics, which is critical for a disease like ALS with limited number of eligible subjects and resources. Alternative study designs are accepted and encouraged by the FDA and the EMA, as we heard yesterday, but also other organizations like Health Canada. And I think it's very important for the Americans in the room to really familiarize themselves with Health Canada, because when President Trump is sworn in January, I think a lot of you are going to be coming north of the border. So I look forward to seeing you all then. Our recommendations. ALS clinical trials must work closely with biostatisticians to determine which alternative trial designs is most advantageous for an experimental therapeutic. And it's going to depend on the drug, on the targets, on the mechanism of action. Trialists should consider the experimental compound's proposed mechanism of action and pharmacodynamic effect on the targeted biomarker to determine the optimal trial design. Trialists should refine alternative design strategies with improved understanding of disease pathophysiology. I just quickly want to go to a study in oncology. And yes, there was a lot. Despite what we heard last night, Hiroshi, I agree with you. There's a lot we can learn from cancer, and I hope we reach the point where cancer is in the near future. And this is an important study published in 2013. It's a negative trial of anaplastic oligodendroglioma comparing radiotherapy, which was standard of care, plus or minus chemotherapy, PCV chemotherapy. And we can see this is a very familiar curve that we all know in ALS. It's a negative study, 300 subjects. Median survival was about a little more than 4 years. This is radiation alone. This is radiation plus chemotherapy, non-significant p-value. However, what they didn't know was while the study was going on, an important discovery was made where they understand that a new understanding of the pathophysiology of oligodendroglioma was uncovered, where if you have a deletion, if the tumor has a deletion in chromosome 1p and 19q, that makes the tumor incredibly chemosensitive and much more likely to respond. So wisely, they banked all the tumor tissues in the study, and once the study was completed and negative, they went back and looked at those patients who had a 1p 19q deletion versus those who did not have that deletion. And this is what they found. They found a dramatically positive effect, whereby patients who don't have this deletion, excuse me, their median survival was under 4 years for patients who don't have the deletion. But for patients who do have the deletion, they're extremely responsive to chemotherapy, and the drug actually works. The p-value is highly, highly significant. Despite much fewer patients, remember, it's a subgroup analysis of this, so despite much fewer patients, they saw a dramatic effect of their drug. So in other words, a study that was negative actually was positive if you would have enriched design for patients with a 1p 19q deletion. And this is why I think this talk perfectly follows the biomarker talk, because we have to know what to enrich for, and we have to have a better understanding of ALS pathophysiology before we can catch up to cancer and have meaningful therapeutics. Thank you.
This is a very important presentation. May I ask first, excuse me, this is what happens in Japan. We have selected out those responders. Early in the course, for example, in the trial of the high-dose method development. But the authorities asked for the second trial with early stage ALS to do active and placebo. So nobody wants to be placebo, and then we are stuck up. And so in that cancer trial, did the authority approve as it is, or required another trial with active versus placebo? That's my question.
So, number, I think exactly you did the right thing. I mean, it's almost naive to think that a disease with this type of phenotypic and genotypic variability is going to respond uniformly to the same therapeutic. So you're right. In the early phases of drug development, you want to try to tease out those responders, and then for the later phases, you have to enrich your design, which is what they did, using patients in the earliest phase, right, of disease for enrichment. But you're right. You may need a second study to validate that. And that's the challenge. That's the real challenge. That's right.
So the second graph to the right, that was a group with a placebo and then drug. Everyone had the gene marker. Everyone had the gene marker on the thing.
And then what this graph is they took all the patients who have the 1p 19q deletion, and they compared those with radiotherapy alone versus those with radiotherapy and PCV, and they're exquisitely chemoresponsive. So dexpramipexole, take all the patients who, for example, have a C9 mutation. I'm just making this up. And then you look back at the data and you tease those patients out and say, wow, dexpramipexole actually works in patients with C9. Doesn't work in patients who don't have C9. So
I think his other question was that enough for regulatory approval?
Yeah. And obviously, we'd have to check with the regulatory agencies. The nice thing about this study is, and we heard it yesterday from the FDA, is look at the p-value. Remember, they're talking about p-values of 0.05. What are you going to do with 0.05? But less than 0.001, that's pretty dramatic.
So nice talk, Lauren, over here in the green. So this idea that we might go back and learn something later that could influence a past trial, how we look at a past trial, I think it's a really important one. And it underscores the need maybe for us to start using the GUID, Globally Unique ID, in all of our trials. I haven't seen that as a recommendation in anybody's subgroup yet, but I think that should be in there someplace. Because if we tie all of our trials together, then someday we might be able to go back and look at some of the other ones and understand them better.
I think that's a great point. First of all, Richard, I'm sorry for missing you. I don't know how I missed those pants, number one. Number two, I think you're 100% correct. Imagine if they didn't bank their tumor tissue in this study. They would have been kicking themselves. And now, when they do these studies, they enrich their design specifically for these reasons. So we will find the biomarkers, and once we do have those mechanisms, that's exactly what we need to look back and see.
So, looking at that example, which is a fantastic example, can you turn it on its head? As opposed to saying, now I know what the biomarker is, go back and re-look at the data. If we do trial designs where we actually define a biomarker after we pull out the patients that actually that did a little bit better. So if you took that same data and if they looked at it that way, could they pick out the patients with that mutation as opposed to going back and, because we don't have a biomarker, they do, but we could actually define a biomarker if we actually looked at those subsets of patients that did a little bit better than the patients who didn't.
Yeah, I think it's a great thought, and I think during the dexpramipexole study, all of us had patients who said, no, I was responding, I was doing well, the drug worked in me, and what was it about those patients? It's just, with so many variables to look at, it'd be very hard to tease, but I think you're right. If in any given trial, there's a good chance that there's a subgroup of patients that are responsive. It's just figuring out what those mechanisms are is the real challenge.
Thanks, very nice talk. I wanted to comment on the Parkinson's experience using historical controls. And what we did was we had planned to use a historical control. We did this in a couple of different trials, but we included a small, as you mentioned, what we called a calibration group, which essentially was meant to check that our assumptions about the historical control group rate of progression were correct. What we ended up with was actually the worst of both scenarios. So it turned out the calibration group, the calibration placebo group, didn't look at all like what we thought the historical control group should. So, but the calibration group was too small to really serve as a true comparator. So we ended up finding out that we couldn't use the historical control as a real comparator, and we had too small of a real control group to use that as a comparator. So in the end, I would say be really cautious about that approach. What really looks like it happened was that the standard of care just sort of changed in ways we didn't quite anticipate.
That's right.
And that caused the shift. But the issue is you never know when that's going to happen. So again, just some caution from our experience. I wouldn't do that approach again.
But I guess it was a good thing that you found that, because then you would have been using a historical control that was not representative. Right?
Absolutely. And I think the message for us was historical controls are often really not representative.
Yeah.
So that was, I agree with others. That was, these examples was really good. And the idea of using the GUI numbers that Rick mentioned is excellent. It does bring up the issue of, in the future, if we could, and I know this is a big if we could, if we had essentially genetic data, biological data, that we would be able to go back, we had stored patient's data, and you go back and say, oh, look, this patient has X, Y, and Z mutations, and you lump them all together, you'd be able to fast forward these kinds of results in the future. I don't know if that's possible. We do bank a lot of material from trials nowadays. A version of that, actually, and Don John's mentioned this, is in the future, there's a big effort underway in the United States to build iPS cells from like 1,000 patients and to be able to identify what those molecular subtypes could be, because that's what you want. You want to be able to go back and do that in patients. So I really applaud that. But, and then unrelated to that, Ben, you mentioned this issue of would that be acceptable to the FDA? Granted, this was an experimental drug, but do we have to worry always about that? If we had a drug that was already in the market, we do this all the time as neurologists, do we care if it meets their approval as long as we would see an effect? If aspirin did that, who cares what the FDA says? We'd have aspirin for a subgroup.
100% right, Jeff. That went from 4 years to 12 years in terms of median survival. I mean, and of course, we would be putting our patients on those medications. And you're 100% right. Those 10 to 15% of ALS patients who are slow progressors, maybe there is a genotype that we may be able to identify one day.
So quick question to clarify on the 1p19 deletion. Was this subgroup marker part of a hypothesis-driven selection of a targeted panel, or is it a non-hypothesis, more empirical discovery?
Post-hoc subgroup analysis. This is after the study. Yeah.
Post-hoc, but there's two ways to do that, right? I mean, you can have a select list of potential response predictive markers based on our understanding of the disease biology, and then you can have a non-hypothesis-driven wider array of analytes to look for.
Right.
And then using machine learning and computational biology, you can select the ones and fit a model to it. So I was just curious if you could.
Yeah. Obviously, the mutation was completely unknown when they started the study, but really, in my mind, it comes down to effect size. That effect size is so massive that you can't help but be immediately impressed by it. But you're right. I think that's the kind of thing that's really important, given we have such a heterogeneous disease.
So just for my dumb clarification, this 1p19 deletion was already a suspect. Or was not already a suspect?
When the trial started enrolling, it was not. It was unknown. It was undiscovered.
Okay.
Afterwards, yes.
Okay.
Afterwards, yes.
Lauren Merritt here. Wonderful talk. You gave like the best example, but there's lots of examples of these post-hoc analysis that then when you repeat the study fail. And so I thought it was just worth commenting on that. Is here you have a biology that can make sense. It can be proven. But often in our ALS studies, when we do these post-hoc analysis, we don't have that. For example, in the dexpramipexole phase 3, we did go back with Knopp and others to look for subsets, and a subset was found that might do better, but there was no biology there, and it really fizzled there. And a lot of the statisticians were kind of telling us, well, if you look, 1,000 times for something, you're going to find something. So I think to answer kind of the methylcobalamin question, I think we can't look at these post-hoc analysis and decide it's ready to treat anyone unless we really understand the biology, and then often you do have to repeat the study.
Absolutely.
Okay, thank you. We have to move on. The next question is question 2. What is the idea of phase 2 trial?
This is Steve Shelsa from Mount Sinai in New York. We were asked to define what the ideal phase 2 trial is. Of course, the ideal phase 2 trial would be to find a drug that stops the neurodegenerative process and reverses weakness. But since until that happens, and because a much smaller effect size is expected, we came up with the following recommendations. Just some background. Phase 2 trials are part of early drug development phase. It's the learning phase of drug development. It involves gathering information instrumental for designing subsequent efficacy trials. There were several types of questions raised in a phase 2 trial. We want to assess optimal dosing, determine the biological effect, monitor safety and tolerability, assess target engagement, and clinical efficacy is difficult to determine, and this is largely because of things that have been reviewed in this meeting, the heterogeneity of ALS and the small effect size expected from the drugs that we've been testing and plan to test. So it should not be, so because of this, clinical efficacy should not be the main goal or objective of a phase 2 trial. It may be possible to design phase 2 trials to assess preliminary efficacy, In terms of defining a duration or sample size, I think that for a phase 2 trial, it will depend on the objectives of the study and has to be individualized. Sorry, this wasn't supposed to be here. Okay. Recommendation 1. Rationale: a biological assay can be developed to determine the investigation therapy being tested that produces the desired physiologic effect. So this can include serum and CSF levels, testing in iPS cells, and neuroimaging. And clinical trialists should consider including potential biologic assays in the phase 2 trial design to determine the relationship of the dose with the physiologic effect. Generally, we'd want CSF levels to show that drugs get into the CSF, but for compounds that are highly lipophilic, that may be less relevant. So then other markers such as neuroimaging may be important. Rationale 2, a phase 2 study should ideally have a measure to show target engagement in the central or peripheral nervous system. CSF drug concentrations may be considered to optimize dosing. Neuroimaging may serve as a biomarker to serve target engagement in the CNS. So recommendation 2, in phase 2 trials, clinical trialists should consider including pharmacokinetics, CSF, and serum and plasma, or at least an appropriate biomarker or biological targets, biological assays, cellular assays, or neuroimaging to show the drug impacted its proposed biologic mechanism and target. And rationale 3, reproducible quantitative strength, respiratory and functional measures are important. Adequate training when measuring clinical outcomes is important, and this is one of the goals of the phase 2 trial, is to prepare and to make sure that people are trained and that the outcome measures are working and consistent. So adequate training is important. Patient positioning, evaluation times, cost, and training are considerations. Assessing safety and tolerability of an investigational therapy is an important objective. Monitoring of drug safety should be consistent. Recommendation 3, clinical trialists should include adequate training for assessments and safety monitoring. Rationale 4, outcome measures. When we were given this question, we didn't have all the questions that were being asked of the conference. So if some of this is redundant, it could be excluded. But for outcome measures, they should be sensitive, reproducible, and validated. It's to evaluate safety and preliminary efficacy. An accepted functional measure that allows for remote collection of data is desirable. For example, the ALS-FRS. Respiratory measures such as forced vital capacity, slow vital capacity, or SNP are also important. Phase 2 trials are typically underpowered to assess survival differences, and an ALS-specific quality of life scale has been shown to be insensitive in that it does not decline with disease progression. Although, as discussed earlier in this meeting, I think quality of life scales may have a role, particularly in showing that a decline of the quality of life scale with an active drug is, possibly is not a good thing for the drug. And so recommendation 4, clinical trialists should consider evaluating preliminary efficacy with appropriate outcome measures. And that's it. So I'm happy to take questions.
Thank you. Any questions? Yes, please.
I'm a bit surprised by recommendation 2. That in phase 2 trials, clinical trials should consider including PK. Shouldn't that happen in phase 0, so to speak of?
Well, I think in a lot of ALS trials, the early phases, phase 1 and 2, were combined in one trial. So I think it would apply more to that.
But without knowing the PK of the drug, would you go to?
Well, I think it's just to further define it, not so much to the preliminary.
So just on a Hardiman, in recommendation 4, not sure what you mean by a specific quality of life is insensitive. Insensitive to what and compared to what?
It's insensitive as a measure of disease progression.
Okay, but it's not a quality of life measure is not a measure to assess disease progression. It's to assess quality of life. So that should be clarified.
Yeah, it's possible we can exclude the whole outcome measure section since it's discussed elsewhere at the meeting.
Marcus Weaver, Switzerland. What I see is that there is a lot of overlap with the biomarker issues. And can you go back to question 2, please? Now, in the rationale, you say that neuroimaging may serve as a biomarker to demonstrate target engagement. I mean, so do you think this imaging biomarker is ready to be used as the imaging is ready to be used as a biomarker? Because we did not feel that we are there yet.
Yeah, I agree. I mean, it was just more of an example of what type of marker the goal of the phase 2 is to show that the drug has some biological effect that's proposed and some measure of that, and that it's reaching target. So I think in a phase 2 study, a lot of what things we choose to show that biological effect are going to be novel things. They're not necessarily established ones.
Mary Kay Floater from NINDS. For recommendation 3, I really think that should be a must, that people must be trained in the assessments.
Okay. I have no problem with that.
I just had a question on recommendation 4. I realize that textbook phasing of trials states that we're not really looking for efficacy in phase 2, but how do we prevent ourselves from going to phase 3 multiple times only to end up with a negative study? I think we need to have a paradigm shift and think about what do we do to make sure that our phase 3 trials are going to be successful going forward.
Well, I think that's something that's been addressed repeatedly at the meeting, and that one of the main problems we have is the heterogeneity of ALS. So, and I personally think that we should be more inclusive with patients when we do prospective drug trials, and then subsequently analyze various subgroups and see if there are subgroups that have a greater effect with the drug.
So I found a lot of good comments and just operational comments. This afternoon we are going to have breakout sessions, and from yesterday's, today's, all members involved with each sections write it down, and we have to improve those guidelines. Tomorrow morning we have a series of just guideline recommendation, no more background, no more rationale, just guidelines one after another. So please improve guidelines with this afternoon's breakout session. Thank you.
I have the last question from the day is very important because now just facing the heterogeneity of the ALS, the role of phase 2 is to find out responders as you recommended in the last recommendation. So that's a change of the paradigm.
Yes.
Any other question? Then you have a question? One last really brief question, please.
Yeah, so I have a question that ties, I guess, a lot of what you talked about, which is more the philosophy of phase 2, which ties in with the efficacy endpoint. And I'm the statistician, so I'm the one, right, that always has to be like the downer in the room, or the doubt or whatever. But it's almost like that that we approach phase 2 wrong, right? We approach phase 2 assuming things work, and we want to see that it works and move forward. And I mean, given the history of neurological trials, we should probably approach phase 2 assuming it doesn't work and saying what evidence would we need to change that to go to phase 3, right? Which gets to the rigor, like with whether it's an efficacy endpoint, it's a biomarker or whatever, is I think when you look at a lot of failed phase 3 trials, you look back at the phase 2, and the phase 2 were designed in a way that they were probably going to go forward regardless. You know, that they're underpowered to show an effect. If there's no effect, you're still going to meet a threshold. So with all the things you've brought in terms of designing that, how do you, I guess the question would be maybe to flip it from what we've talked about with the heterogeneity and how do you get promising things forward. How do you approach phase 2 design, at least in the way that you viewed it, such a way that if things are not likely to work in phase 3, you would not go forward to phase 3, which is a slightly different approach to the problem?
No, I think with the phase 2, because of the heterogeneity of the disease, and you may see trends of efficacy, but you may not. And I think if you don't, we may be excluding compounds that still have potential. So I think that's why it's important and why we emphasize that you have some sort of marker to show that the drug is reaching target, that it has the desired biological effect, whatever assay best shows that can be developed, and move it forward for that, you know, in that way.
See that Chris and I are dressed the same today. So I'm filling on the next stage of the stats review. I think maybe to operationalize it, I would propose that you consider a recommendation of defining explicit go, no-go criteria prospectively, not so, and demonstrating efficacy is probably not a reasonable criterion, but there should be some pre-specified clear decision rule that could be applied to whatever data is going to get.
No, I think that's a good point.
Okay.
Let's move on to question 3. Ryuji, and please. What constitutes positive phase 2 result? This is another big question.
And forward is this one. So it keeps on flipping. Okay. So our question here is what constitutes a positive phase 2 result? And our background is that we have to deal with when do we move into that phase 3 study, as has been mentioned in some of the preceding comments. And one of our challenges is that phase 2 are not typically powered to really look at efficacy. And so then we have to determine criteria. What's the bar? Where are we going to set the bar to move forward to a phase 3 study? If we set it too low, we end up with a lot of phase 3 studies that are failures, and we've seen certainly a number of those in ALS. And if we set it too high, we may end up missing a compound or an intervention that would be beneficial. What are we good at looking at in phase 2 studies? Well, we're good at looking at safety and tolerability. It's a good way to look at dosage selection, and it may also allow us to see whether or not the intended target has been engaged. So our first rationale is that phase 2 studies should, since they are a way of looking at a compound or an intervention typically for the first time in large numbers of patients with ALS, pay a lot of attention to adverse events, and tolerability for the compound or the intervention should be a priori defined. positive results for safety and tolerability are one of our major goals in supporting moving towards a phase 3 study. So our first recommendation, clinical trials, and in the spirit of the Delphi method, we'll drop the can and just say should evaluate safety and tolerability endpoints with statistically and through the lens of clinical relevance, and that safety is often semi-quantitative and should take into account the frequency of adverse events, including lab abnormalities. For our second rationale, we know that dosage selection is most applicable to drug studies, but in intervention studies, there may also be something along the lines of timing to take into account. So selecting an appropriate dosage for phase 2, phase 3 testing is a primary aim of what we're trying to establish in these phase 2 studies. So our recommendation 2 is that clinical trialists should select the dose for phase 3 testing based on pharmacokinetics, pharmacodynamics, and/or safety and tolerability. And that positive phase 2 results are established by the successful selection of an ideal dosage for phase 3 testing. Our third rationale is that and we've talked a lot about biomarkers today, so this is one of those things that does show some overlap with some of the prior recommendations. So we'd love to have a biomarker that everybody accepts and is well established, and we all know that that doesn't quite exist yet. But certainly including biomarkers that have been statistically powered to show change would be desirable. And since we don't have these biomarkers for ALS, our recommendations are as follows. Clinical trialists should include a pharmacodynamic biomarker or measure whenever possible. And exploratory biomarkers, while encouraged, need to be interpreted cautiously. For the fourth rationale, clinical efficacy outcome measures are often included in phase 2 trials, yet these trials are not powered to detect frequently statistically significant change. So we look at ALS, FRS, and slow vital capacity, and HHD, but we may not have the number of patients to allow us to detect statistical significance, and rarely are phase 2 trials going to be powered to allow us to look at survival. So clinical trialists, as part of our recommendation 4, should view clinical efficacy outcome measures as potentially informative, but positive only when clinically meaningful and statistically significant or demonstrative of a strong statistical trend toward benefit. And we're open for discussion.
Thank you, Stacy. That is exactly the point of a phase 2 trial. And too often we make expectations of phase 2 trials that somehow they're going to give us some positive result that's going to allow us to go to phase 3. And we see that in the literature all the time where small trials are over-interpreted to suggest that there is some clinical benefit, or in fact that companies or groups only decide to go to phase 3 if there is a, quote, positive clinical effect in phase 2. But phase 2 trials are not designed or powered to do that. And I think that's where we run into a lot of trouble. We have a lot of enthusiasm for phase 3 trials sometimes because it said, oh, look what happened in phase 2. But in fact, the phase 2 is too small to really tell us anything except safety, tolerability, and dosage. And that's what a phase 2 trial is all about, and I think we just have to accept that. So I think your group has hit the nail right on the head.
Thank you.
I have a slightly different opinion on this. So we're talking about a paradigm shift. We're going to be drafting guidelines that will stay for 3 years, maybe 5 years, maybe longer. So we're using the criteria of tolerability, dosage finding, and engaging a pharmacodynamic target, a biological target as criterion to move into phase 3, but obviously this still may not be enough. If there is no clinical signal of efficiency, there's a high likelihood that the phase 3 will fail, and that's expensive. So the question comes back to the biomarker discovery, I guess. So I wonder whether we would be able to put into language, into the recommendation, that if a biomarker in the next few years is discovered that, we can't use the term surrogate, okay, it correlates tightly with a functional outcome measure, can you use this in phase 2 to reduce the sample size and do a preliminary efficiency analysis from the phase 2 and use this data as critical endpoint to decide whether we're going to move into phase 3 or not?
I agree that in an ideal world that biomarker would provide that. It's right now we just don't have it. I think you want to see some hints of efficacy. You certainly want to see that the drug is well tolerated, is safe, and things are at least moving in the right direction. But particularly in a 2A study, you know, the first in disease, you're just not going to have enough data to really expect to see that much. Other questions? One more.
It's over here.
Hi, this is Ed Tesoro again. I want to bring this big question of the time frame of phases 1, 2, and 3 back to the forefront, because just from a patient perspective, as many elements as we have to assure to go 1, 2, and 3, those 4 years, the ALS patient on my left and the ALS patient on my right won't be here anymore. So I know that the way it's built, as John just said, phase 2 has specific guidelines, but can we wait all the way to phase 3? I'm in my 4th year in this trial, and it's a miracle that I'm here, you know, statistically. So shouldn't we be asking, not just to meet the criteria of trial dynamics that exist, but shouldn't we be forcing a 1 and 2 in the same time frame, so that by the time we get to 3, it's not 4 years down the road? I mean, as much as I love the scientific method, in this particular disease, do I have the luxury, or any of the 30,000 of us, to watch 4 or 5 years go by? Because it's never going to be about the people in the trial. It's going to be about another generation, which is fine. But is that the best we can do? Or with this particular disease, should we say, if we have to do 3 trials as currently devised, then, you know, I'm thinking of Dr. Vickers last night saying he can't believe that. Now, of course, he's a oncology surgeon, so it's a different game, I understand. But shouldn't we be challenging to go to 2 phases, combining the first 2 and then going to efficacy?
Well, occasionally some studies are combined 1, 2 or 2, 3, but remember that phase 1 has a different, you know, there are different goals to be established by each of those phases, and hence that kind of forces us in some ways to do phase 1 trials separate from 2, separate from 3.
Well, I know that's the current dogma, but is this meeting for these 4 days one that should say, is that current dogma one we ought to attach ourselves to? Or, as a number of people have said this week, should we be looking at a new paradigm, because it's not like what we've done in the first 15 years has been great? So should we really be stretching ourselves to try to get shorter, better, and maybe, and I know it's a big subject, a layman can easily ask a question like this, but I'm wondering if anybody else has my same concern for all that.
Thanks. I just comment, I think it's an excellent point. Later this evening, I'm going to talk about gene therapy and some relevance to cell therapy approaches. I think sort of 2 studies, you know, an earlier one focused on delivery and dose selection, and a later one focused really on efficacy. That paradigm, but I think it may be very treatment modality specific. What we've been talking about is a very small molecule traditional drug development approach. And what I wanted to comment is just the way some of the recommendations are going to fit together, I think will end up yielding a very high phase 3 failure rate. And maybe that's okay. Maybe that's okay because you want to make sure that you haven't missed anything. But I think that's what will happen. What we heard from biomarkers, and Michael articulated, is really there are very few things that you can measure well. And the recommendation here on phase 2 is to use some of those biological measures, and that phase 2 isn't about looking at clinical outcomes, but about safety and tolerability and, if possible, dose selection. But I think what we're hearing is there isn't a whole lot of assay or measurement substrate to work with for dose selection. So you're going to end up with a phase 2 that says, well, it was safe and tolerable, with very little evidence that there's some activity. The best way to get a safe and tolerable drug is one that doesn't actually do anything biologically. So I think, another framework, and maybe ask some of the statisticians to comment, is to think about phase 2 conceptually as a diagnostic test. So we all know there's some prior probability of success before you ever start the trial, and based on historical experience is pretty low, right? It's probably in the 5 to 10% range. So the question is, think of it as a diagnostic test. What's the probability or the positive predictive value after your phase 2 that it's you actually have a drug that works or not? If you look at a lot of different designs, you'll see that they don't move the needle that much, and the positive predictive value is extremely low. So I think, we have to be cognizant of how these recommendations fit together and what they'll really yield in a drug development pipeline.
Tipu?
I'm sorry, we are running out of time, and sorry, we have to move on. And the last part is review of the Old Guideline.
Wait a minute, we have one that way. Do you want to go through the questions? Hold on.
Yes, we have to adjust the slides just a second. This is going to do the review of the prior recommendation.
Okay.
We're going to do the review of the Old Guideline.
This is the recommendation 13.
Oh, okay, great.
Yes. Yeah. So that should highlight the fact that this was a new topic for the group, and it's not in your handout, and it's perfectly imperfect, Gary. So, we'll ride through this all together. So we were asked, and I have, I think, a few minutes just to bring up the recommendation 13 from the previous guidelines, which says that phase 1 trials should usually incorporate a concurrent placebo control and should be conducted for 6 months depending on the design. And this was a late entry into our group and created a robust discussion. And there were 2 points that created a lot of discussion among our group. So the first point was the use of concurrent control, placebo control, and the second was the 6-month duration. And so interestingly enough, the prior meeting was in April 1998, and subsequent to the meeting for this clinical trial guidelines publication, there was a publication by ICH, International Council of Harmonization, which actually gave general points of consideration for specific phases of trial. So that serves as the background that's up there right now. So the purpose of phase 1 studies, and the ICH-EA discusses it as studies of human pharmacology, is to really assess the safety and tolerability profile, the pharmacokinetic and pharmacodynamic profile. And the pharmacokinetic profile is essentially trying to describe the drug absorption, distribution, metabolism, and excretion, and really characterizing the preliminary characteristics of whatever therapeutic compound that you're investigating. And the last is to do dose tolerance studies, or determining maximum tolerated dose. And really, the phase 1 studies are to help to determine what dose you'll be expecting to use in later clinical development. I'm not sure exactly what's happening, but
The approach, so there are guidelines both with FDA and EMA that specifically give some guidance to the approach of phase 1 studies. So typically, a lot of the information is gained from the preclinical or non-clinical studies, and there's an expectation or an estimation of risk and potential benefits from those non-clinical studies into the first in-human studies. So they recommend single and multiple dose pharmacokinetic and pharmacodynamic studies, and in addition, dose tolerance studies and drug interaction studies. There's other topics that we wanted to include, which not only includes design, but also the inclusion of controls and which patient populations to study in the phase 1. So the first recommendation is based on just the studies that are included in phase 1 and from the recommendations both from the Code of Federal Regulations and both ICH. So generally we want to understand the dose range, the therapeutic efficacy, not therapeutic, therapeutic safety, and the expected doses to be used in future clinical trials. So we do a single ascending dose study to understand the initial acute safety and pharmacokinetics, as well as multiple dose or multiple ascending dose studies to evaluate toxicity after multiple doses or longer exposure to a potential intervention. So recommendation 1 says clinical trialists should start with a single ascending dose study, first to evaluate acute safety and maximum tolerated dose, to evaluate pharmacokinetic profile and tolerability. And the subsequent statement actually probably should just fit in the background in the rationale. The second concept is the use of placebo control. So the primary objective of the phase 1 studies is to understand the safety profile, and really when a phase 1 study is conducted, we're trying to understand the nature and frequency of adverse events to a potential therapeutic intervention and the relationship to dose. So there is a role for placebo-controlled phase 1 studies. Now, the landscape has changed for ALS, and I think this is where Bernard was making a point about cellular-based therapies and gene-based therapies, which can change how you do a phase 1 study. So what we did was we recommended that clinical trialists may use a placebo control in phase 1 trials to evaluate the relative differences in the occurrence of adverse events between the treatment and placebo groups. But we realized that some interventions involve risk, maybe due to delivery. Some interventions, like cellular-based therapy or gene-based therapies, have permanent effects, and so it may not be appropriate to have a placebo-controlled, or it might be specific to ALS. And so in those particular considerations, trialists may choose not to include a placebo in phase 1 studies. So we talked about study design, we talked about placebo-controlled. The last recommendation talks about healthy volunteers versus ALS patients, which population to study in a phase 1 study. And so we talked about single ascending dose and multiple dose studies of potential ALS interventions being performed in ALS patients or in healthy volunteers. In most cases, involving healthy volunteers for these initial studies reduces risk, since healthy volunteers are more likely to be resilient. Furthermore, enrollment could be faster. However, in some cases, and again, this is reflecting some of the changes in and evolution of the therapeutic interventions being tested, it might be more appropriate to use ALS patients in the phase 1 instead of healthy volunteers. The choice really depends on the expected risk and the potential benefit of the therapy that you're investigating in phase 1. And those expected risks for that therapeutic intervention really comes from the information that's gained from the non-clinical studies. So there are risks in delivery of the intervention. There could be permanent effects, and really the change and evolution of the therapeutic interventions in ALS and other diseases involving cellular-based therapy and gene-based therapy actually has triggered publication of guidance by the FDA specifically to address cellular and gene-based therapy in June of 2015, and also in EMA in March. So in the FDA guidance, they actually state that the benefit-risk profile is not acceptable for use of healthy volunteers in cellular and gene-based therapy. So our last recommendation here says clinical trialists may conduct phase 1 studies in healthy volunteers first, but if there is a compelling reason and really is an understanding of the benefit and risk, it may be more appropriate to include people with ALS in phase 1 trials. And so that's our last recommendation and update for that.
Because of time, can we take any question?
No, just
No question.
So that's it, right? Good. Thank you so much. So we have now coffee break. We get together 10:05. Thank you.
Oh my God!
I want to talk to you about it. We're in the same situation. We have problems.
Yes, I did. You guys look good. Y'all pretty good.
Modify your slide as long as you can go to
Oh, really? And you took him as an undergrad?
Yes.
Oh, of course, contracts.
Thanks.
The local pharmacy in this other pharmacy. So like a funding coordinators, personnel will have to be our, yeah. Yeah, of course, I understand. Oh, nice. So the labs are going to be done in the insurance here, right? Yeah. Good, good. But the shipments is going to be your... Okay, good. You're not doing anything, or you are. We could do. I think it's realistic. I mean, if you're going to give one or talking and you can play from your experience, that's not going to be sick. Yes, they can. Yeah, but you don't mind having something. Yeah. Oh, that's good. So, you know, we can do it. I mean, as long as we find one. Right now,
Ah, good. Same moderator.
Yeah, I like that. No, it's
Yes, I do agree with consistent assistant David. That was actually surprised that he wasn't the link out himself. So that's right. We alternate. I mean, the guy does pick one and three, one four, but he eventually was able to arrange a week that my colleagues do, and they follow ups. I see the news. So I was like, we both are there. It's busy, but no, but it's even true for us. That we haven't been anything that well. The second one. But we agreed that operating research. Yeah, one of the universities where I work, and another one how each among 150 people. So, yeah, I know we set up a mutual agreement in terms of research and exchange. They want to continue those labels and extension. It's a pill 2 times a day or yeah, we
have to incorporate suggestions, comments into new draft recommendations. Recommendation has to be improved. And also I want you to look at anything deficient, anything overlapping other areas. You're talking about number of overlapping. And you may need to talk to other groups to how to consolidate too many overlapping. Certainly recommendations should not have too many overlap. And also I want you to think, why is something deficient? Why is something, that sort of thing you need to once again discuss with your group session, breakout session this afternoon. Tomorrow morning, we're going to present all recommendation one after another. So brief presentation and then brief discussion with a lot of recommendation tomorrow. So recommendation tomorrow should be improved compared to today's recommendation. So we're going to start, and Matthew Kiernan and myself. So just lead this session.
Well, thanks, Hiroshi. We're just hearing actually in the coffee break all the work that Hiroshi has done to put this together, and it really is a phenomenal effort. So it's a great privilege to be up here with him. So I'm just going to continue on now with the different trial phases, and so the next part of it is how do we make decisions to move from phase 1 to phase 2.
Great, thank you so much for this opportunity to present our work. This is on the question, how do we make decisions to move from phase 2 to phase 3 clinical trials? And I have to say that as a group we really grappled with some of the issues that came up when discussing this question. So what I'll present to you today is a consensus opinion from the group. In fact, I'll be reiterating much of what's been said by my colleagues in Group 4A in the previous 3 questions. So I'd just like to start with some background to this topic. So we know that phase 2 clinical trials are geared primarily towards gathering information to optimize the design of the subsequent phase 3 clinical efficacy trial. So really, in phase 2 trials, efficacy has in general not been the primary aim of the study. And that's for a number of reasons which we have discussed earlier today, namely that firstly, they're often vastly underpowered to be able to assess clinical efficacy. Secondly, they're often not long enough in duration to be able to make a judgment on efficacy. And thirdly, as we've heard repeatedly over the course of the last 2 days, ALS is a very heterogeneous disease. And so that compounded with the lack of power mean it's difficult to make a good judgment on efficacy measures. And so with this in mind, the key questions that should be addressed during the phase 2 trial are threefold. So firstly, what is the safety and tolerability of the intervention? Secondly, how well can we choose a dosage to take forwards to phase 3? And thirdly, is there evidence that there's adequate target engagement or a biological effect? And so these three key questions are very intimately linked. The dosage that's selected to take forwards should maximize the target engagement or biological effect whilst minimizing any issues with side effects and tolerability. And in terms of looking at target engagement, ideally we need pharmacodynamic biomarkers which can tell us whether that intervention led to an actual biological response in the patient that was treated. And we would argue that ideally all three of these questions should be addressed as far as possible at phase 2. And this is to prevent us going forwards into very costly and time-consuming phase 3 trials. It may be that we need to conduct further studies at phase 2 if any of these questions remain uncertain. So against this background, we have two major recommendations for this question. So I'll take them in turn. So the first one is based on the rationale that, as I've said, phase 2 studies tend to be highly underpowered to be able to detect a clinical effect. And so, nevertheless, we do often include clinical outcome measures at phase 2, whether they be primary or secondary. And so in this case, we recommend that clinical trialists should only use clinical efficacy to support the decision to move from phase 2 to phase 3, when firstly, the clinical outcome is stated a priori, and secondly, the biological effect is large, is plausible given the known mechanism of the drug, is robust across all patient subgroups, and is shown to be statistically significant. So moving on to the second recommendation. So before moving to a phase 3 study, it should be well established that there is adequate data to understand the safety and tolerability of the intervention, to be able to optimize the dosage that's taken forwards, and for evidence of target engagement or a biological effect. As I said, this is down to pharmacodynamic markers of target engagement, and this is looking in particular at what type of target tissue we're aiming for with the intervention. So it may be that we need a pharmacodynamic marker in, say, the central or the peripheral nervous system, depending on the intervention. And ideally, we would like to have measures of the CSF drug concentration if the intended target tissue is the central nervous system. So against this rationale, our second recommendation is that clinical trialists may move from phase 2 to phase 3 studies with, firstly, adequate information on safety and tolerability, secondly, information regarding pharmacodynamic optimal dosing, and thirdly, evidence of target engagement. So those are 2 recommendations, and it's open for discussion.
Well, thanks very much. Very clear. So open up for any discussion.
I would say, so this is, I mean, this was something I think that we talked about a lot as a group. It was very well presented. Thank you for taking that forward. One of the things that we've struggled with or talked about at various points in the in the meeting over the last day is that there's a tension between saying what we can do today and what we'd like to do. And I guess this, in refining the guidelines, one of the things that we'd like to know as a group, as our subgroup from the larger group, is that we lay out a pretty high bar to go from phase 2 to phase 3 here. I don't think that my understanding of the way that people might use trial guidelines like these would be to say, the guidelines say, really, ideally, you should have safety and tolerability information, pharmacodynamic information about an optimal dose, and evidence of target engagement. A trialist, don't actually have that. It doesn't mean you can't. My understanding, and I guess it's sort of just a question for the group. My understanding would be it wouldn't mean that I, as a trialist, can't take my product forward, but that I should have fairly good reasoning to say why I haven't met those guidelines. And I just wondered from the group whether that makes sense. Does it make sense to set a high bar like this? What are people's thoughts about it? This was a big point of discussion in our group.
Let me interject. This isn't a high bar because it says may.
Oh.
Yeah.
So, no, that's important.
Sorry, I took back up.
You need to say should?
No, I mean I think we can say may. I think may is the right word. On the other hand, I think though we know the nuances of may and should here, I think it could be read as more than that from an uninformed, from somebody who hasn't sort of studied the may, should, must paradigm. And so
So should means usually. And we'll make that clear in the manuscript. May means sometimes. It's okay.
Yeah.
Must means always. And so it sounds like you were describing usually you would want to meet these criteria. There may be exceptions, so should would be appropriate.
I'm afraid that I fear that the reality is that may has to stay because that's where we are with drug development. But that a higher bar would be should, and we can't say must. That's really the question.
Add to that.
Could I just make a comment, which is to say that number 1 is not a big problem, safety tolerability. In number 2, we only have half of it now. We can see what the optimal dose is in terms of safety, but in most instances, we're not able to see what the optimal dose is in terms of target engagement and efficacy, which is also true of number 3. So just telling it like it is, all of us who do phase 2 trials realize what we're up against. But more than ever, we are now setting a higher bar. And this is, to me, what this is all about. It isn't pie in the sky. We are engaged in the root. And so I think we ought to state the goal this way.
Great. Yeah. Yeah. Further comments? Yep. I just turned the mic on.
Just turn.
Yeah, you must turn it on. The microphone, that is.
David, so one question I've wondered about is you've been very appropriate in reminding us that these are not designed to demonstrate efficacy. But what about, and has there been some discussion about clinical trialists may consider using a futility design and proceed to phase 3 only if it does not demonstrate likely futility, or however that would be phrased. I find that design appealing, and I'm interested to know what the experts on this think.
Well, I think as we discussed in the first question within this subgroup, there's definitely a role for alternative ways of designing these studies. And of course, we're considering just in isolation, how do we judge phase 2 studies that will then move to phase 3. But absolutely, I think there's a role for thinking about adaptive phase 2 to phase 3 studies and interpreting. I think the most important thing is that we can assess how far we've got the data to look at these 3 key points, because it's very difficult to then move to phase 3 and then make a judgment on, let's say, a negative phase 3 study. Is that because we just didn't engage the target? And so I think that these are sort of the most critical points to look at phase 2, and that absolutely there's a role for more adaptive designs and different trial designs to really narrow down the time frame going from a potential target all the way through to a phase 3 study.
Thanks, Alex, and got some more answers.
Thank you, Matthew. So I was wondering, while we're all thinking about, going to smaller clusters of populations, or even ultimately, trial of one, we're still thinking about bigger phase 3 with hundreds and thousands of patients. And it seems that whatever we're discussing here does not fit that, future model or even current model where we're administering, I don't know, AAVs or stem cells or whatever targeted to a person rather than huge populations. And I wonder if it's, here the guidelines to stay, we may address this somehow as well. And I know that Shafu is going to present something on this, but it seems that when we're discussing phase 2 to 3, and even 1 to 2, or even 1 with placebo, right? So you would never administer placebo, or AAV placebo to a healthy population, right? Or
I must use the microphone. So the FDA guidance does, there's a specific EMA and FDA guidance on the cellular and gene-based therapies, and I think now as the discussion is evolving, James and I realize that from phase 1 to phase 3, the design approach, the analysis of the data that comes out will all really depend on the intervention or therapy that's being used, and probably the guidelines will need to reflect that, because that's the current state now and likely to evolve in the future. So I think we're trying to make it so that it won't be outdated next year. But I think when we go forward, I think we'll try to tease that out in the recommendations.
So my comment does relate slightly to Alex's remark about moving forward in defined subsets of patients. We've been talking a lot about post hoc analysis and looking for responder groups. So this actually goes back to recommendation 1 as it relates to the go, no-go determination part of a phase 2 study. And what I thought the most profound part of recommendation 1 is something that's very consistent with best practices, we would all agree, and that is the a priori determination of the outcomes we're looking for. So in billiards, you can't, or you have to call your shot, right? You can't just accidentally knock the 8 ball in and say you wanted to do that. So that's very clear, but what I would like to know and maybe suggest as some language is, okay, where do we go from there? So we didn't overall meet the study objective, but we want to somehow establish what the targetable population might be for a phase 3 study. So we have our defined subgroup in a 2A trial or maybe a 2B, 2B1 trial. Do we then validate that in a second phase 2 trial? I mean, I would expect yes, but I'd love to see some language about the actual path, maybe in the form of an hourglass where we have a wider inclusion and then we define a subgroup from that data, gets a little narrower, then we expand upon that in a phase 3 trial after validating the subgroup.
Yeah, I think that's a really important point. I think it draws on much of what we talked about yesterday. The difficulty is when you're making those sorts of stratifications into smaller subgroups, do we actually have the power to make a judgment on clinical efficacy? I think at phase 2 in the sorts of studies that we're talking about here that may have less than 200 patients be going for 12 months, it's quite difficult to then sub-stratify and look at subgroup analyses. But yeah, that's something that we can discuss later during our breakout.
A quick comment, and it's sort of back to the nuances of the may, should, must with respect to this particular recommendation. I know James said he sort of prefers sticking with the may, but so my question is, so if you actually had all these, so you actually had safety, tolerability, you had PD on optimal dose and target engagement, would you ever not go to phase 3? So my question is, should, in this particular case, I mean, there can be cases where that is true. And so, so here I would say, could it be the wording be, you should move to phase 3 if you have this? And if you have one or two of these, maybe that's a may. But if you're going to go with these three things, maybe at that point you should move to phase 3. And again, it's all nuances of how we're trying to word it for the guidelines. But so I just wanted to throw it out because as I sat there looking and hearing what you said, if all three of those were met, there would be times we probably maybe still shouldn't for whatever reason, but you probably should move to phase 3. And if you only hit one or two, maybe that's a may. I don't know. So I just threw it out.
That's the subjective of it.
Yeah, mine doesn't have a permissive feature to it. It's a probabilistic. And I tried using it once and then that's no mind.
I never really liked that. Yeah. What we might do is we might, since there aren't any more questions, is there a question up the back there? Or? Yeah. I just put the microphone on. Here.
Sorry, very nice. I'm wondering if you might want to refine what you mean by adequate safety and tolerability information, because I think it's easy to get a false sense of security and sort of declare safe and tolerable when in fact after phase 2 you know very little. And so I think really it's adequate tolerability to test the hypothesis in a phase 3 is kind of really what we mean. And then on the other end, the target engagement, I hear what everybody's saying about you raising the bar, but in some ways I think the emphasis is misplaced of having that in phase 2. If that's not picked up in a phase 1, it probably never should be in a phase 2. And if it's tested, the compound is tested in healthy volunteers, they should have the target, or it shouldn't be tested in healthy volunteers. So I think I might just, you definitely shouldn't be going into a phase 3 if there's no target engagement evidence, but probably should know that before the phase 2.
Yeah, so I think, so when we talked about target engagement as that third point, we're particularly thinking about pharmacodynamic markers in patients, so showing that there's a biological response in the same direction as you'd expect the drug to be acting mechanistically. So it's more specifically looking in the patient population.
Okay, thanks very much. That leads now very well into the next item from Group 4A. What is the ideal phase 3 trial?
All right, so our question, what's the ideal phase 3 trial, which is somewhat of a loaded question, kind of hard to address. In my mind, from the statistician, the ideal phase 3 trial might be different from the way most people approach it. It's not a trial that shows a positive effect. The ideal phase 3 trial design is the one that answers the question. If the drug doesn't work, it shows that the drug doesn't work. If the drug does work, it shows that the drug does work. And a lot of that hinges on the discussion that we've heard from the others. But so what we kind of struggled with was coming up with some broad recommendations, which really had to do with looking at the long track record of failed drug trials in ALS and looking at what can we learn from that to try to think about how to design trials in the future. And so that was really what the focus was on. Although, again, kind of soapbox moment, it's not the design's fault. Unless the drug worked and the design was the wrong design, more likely the design was right, the drug was wrong. So the first recommendation had to do more with, so the rationale was in deciding on phase 3, you really need a panel of individuals from different backgrounds. So the scientists, pharmacologists, clinical trialists, statisticians, clinical experts, to kind of look at the evidence collected in phase 1, phase 2, like what information do you have from phase 2, given the last discussion, to decide where to move forward. So the first recommendation we had was that clinical trialists should obtain input from an expert committee to evaluate the overall scientific basis of and to inform the phase 3 trial design, which is probably what a lot of groups are already doing. The second gets into the kind of the biomarker issue. So the lack of adequate and accepted surrogate biomarkers makes it difficult to evaluate signals from earlier phase trials to inform further development. Biomarkers can help provide early signal of effect. So the second recommendation was the clinical trial should include a biomarker that was evaluated in earlier phases of the development in phase 3 trials, which is kind of an additional validation step. So if there was some biomarker you used to show effect in phase 2, look at it in phase 3 to kind of further validate that the biomarker was well. Or if you don't see an effect, you might determine that it's a poor biomarker for whatever pathway you were looking at, which would be important for informing future studies. So rationale 3 was lack of complete understanding of dose response of investigational therapy may lead to a failed phase 3 trial. We struggled with this a little bit. Our recommendation that we ended up on, so a lot of the discussion here had to do with whether you should require multiple doses in phase 3 or not. And so where we kind of in discussion came down with this kind of a compromise that clinical trials should design phase 3 trials to adequately evaluate the dose of the investigational therapy or multiple doses when the dose response is not completely understood from early phase trials. So it kind of gets to the discussion we had. How much rigor do you need from the phase 2? Do you completely know dose? Do you kind of know dose and you want to do maybe some type of adaptive phase 3 with multiple doses and you might drop a dose and proceed forward? Rationale 4 is that an ideal phase 3 trial should have an adequate comparator, such as a placebo-controlled arm, to assess efficacy of a potential therapy. And so our fourth recommendation, clinical trials should include a placebo-controlled comparator to assess efficacy in phase 3 trials. The fifth rationale is that, again, which is we've talked about a lot over the past day and a half, the heterogeneity of the disease may reduce the possibility of detecting potential benefit in subpopulations of patients. And so our fifth recommendation was that clinical trials should develop inclusion-exclusion criteria that take into consideration the mechanism of action of the investigational therapy, the potential target, and the outcome assessment enriched enrollment. So consider that where applicable. Rationale 6 gets into the sample size issue. So the rationale being that sample size should be based on the clinical meaningfulness of the endpoint and the statistical power. And the phase 2 trials provide an opportunity to evaluate preliminary signals of efficacy and to conform potential endpoints in the phase 3 trial. And so our recommendation 6 is that clinical trials should carefully review the results of phase 2 trials, choose a primary endpoint that's clinically meaningful and adequately powered to detect a meaningful difference, which, again, is a loaded statement where a lot goes into that. I mean, one of the things I think we want to be cautious about is looking at the evidence more broadly, not just taking estimates from a phase 2 trial, which usually have a lot of noise, a lot of variability, and solely using those for your phase 3. And then rationale 7, which I think is an important one that has been addressed kind of in the clinical trial literature as a whole, is that clinical trials that are negative are less likely to be published or even cited, and that publication bias prevents critical analysis of the entirety of research and slows progress. Things like clinicaltrials.gov, that problem is going away somewhat, but not completely. And so our 7th recommendation is that clinical researchers should have a publication committee that's involved in a phase 3 trial, whether industry sponsor or investigator initiative, to encourage the publication of results. So in other words, whether the trial is positive or negative, we want to encourage publication of results. It's important kind of for the scientific community to move forward. So those are our recommendations. Glad to hear questions, discussion.
Again, very comprehensive and so probably Christian statistician. So Ryuji, if you'd like to kick it off.
Very good summary, and I'd like to suggest one aspect on recommendation. All the negative phase 3 trials should have their raw data on the public domain so that we can analyze how it has failed, so that we can sort out a kind of responder group for the next trial or other agents to find out a successful subgroup. And so that's my suggestion.
Yeah, I think that's an important point. I mean, I think data sharing is an important kind of change in the field where now in NIH grants you have to require data sharing. Data sharing is moving into other things. I agree. Things like that will allow you to combine data from multiple resources is important. And probably, I mean, that kind of addresses phase 1, phase 2, phase 3. We should probably be looking at data from past trials in all situations and trying to inform all the phases. Yeah.
I think it also dovetails the recommendations of Group 3 as well, incorporation of biomarkers in each of the studies, which would help that to move forward. Yep.
Yeah, I'm feeling aggressive, so I have problems with the first 3 recommendations. And the first one is that you should involve a committee. I'm not sure that that's what we should be putting into the published guidelines. Maybe a committee is useful, but it may well be that just the group of investigators who are involved should be the ones who make that decision. So let me go through each of the 3 before you respond. The second is that you should include a biomarker. Well, I think we've had a large discussion that there may not be adequate biomarkers available at this particular time, and therefore to put that into the recommendations may not, at this moment at any rate, be justified. And then the third one, to dose range in the phase 3 trial, if you have in fact established an effective dose in the phase 2, may again not be necessary. So I have problems with each of these at this moment.
So let me address them one at a time, because I think we're actually saying the same thing, and it might be language slightly. So for the first one, I agree, and maybe the word panel or committee is too strong. I think the main point there is that you really need kind of a diverse body of people from different expertise, right, providing input, whether that's a group of the investigational team, it's a formal committee or not. So that may be something we can clarify in terms of the language. For the other 2, I think I agree with you, and I think our language that we have in the recommendation addresses that. So for the biomarker one, the recommendation is not saying that you have to include a biomarker in phase 3. What it's really saying is more that if you had a biomarker as an important part of your phase 2, right, whether it's a validated biomarker or not, you should probably include that in your phase 3, right, to get a better sense of how well that phase 2 knowledge you had in the biomarker, how well it informed your phase 3, whether that's a suitable biomarker in the future. I think if you had no biomarker in phase 2 or it wasn't a component of your go, no-go decision, I don't think we're saying that you would have to have biomarkers in phase 3, just for the sake of having them. Yeah. Yeah, that's probably, we could clarify that. Yeah. Yeah, so if we change the clinical trial should include a biomarker if it was evaluated in phase 2, that might clarify. And then kind of the same thing in recommendation 3, and that was one of the things we struggled with, whether you should require multiple doses or not, because I agree with you, and the language that we had in here was trying to clarify that if you feel like you know the dose from your phase 2, you shouldn't require multiple doses in phase 3. And that was where we had should require multiple doses when the dose response is not completely understood from early phase trials. Maybe we can clarify the language. So if it's completely understood, there's no need for multiple doses. But if you have uncertainty on the dose, then you might want to consider multiple doses.
So continuing my aggressiveness, I would recommend that you drop all first 3 recommendations.
2 and 3, I'm not as strong about. 1, I think, is critically important. I think you have to have you should talk to the statistician. Statisticians are good people. Yeah. Yeah.
Well, yeah, I wonder if you should, at this stage, write something about, to differentiation between the impact on symptoms, symptomatic effect, versus a change in the natural history of the disease. I remind that some years ago, the delay start design has been proposed as a way to differentiate between the symptomatic impact from change in the natural history of the disease. So I wonder if we could write something about this differentiation.
I think we could. One of the things I think we're a little hesitant about is to not get too prescriptive here, because I mean, in some ways, go back, not to belabor the point, but the first recommendation is kind of the most critical one, is that you have groups of individuals. And then the rest of it is kind of hard because it depends on the specific drug, the specific situation, what you know from your phase 1 and phase 2. And you kind of have to take all that into account. So if that's the paradigm you're in, that would be important, but it might not apply in every situation, which makes it somewhat challenging to say what's the ideal phase 3, because so much of that will depend on what comes from the earlier phase studies.
Nigel Lee, I feel Wally's pain. It's a measure of how many trials that we've done and failed in. But could you go back to number 1? I really do beg you not to refer to committees. What you need is the scientific input. Yeah, what you need is the expertise. So I think it should be in terms of expertise, not a structure.
Yeah, I would agree with that.
Yeah. I have a different opinion on this, because I would suggest making clear who are the persons or the type of expertises that are represented in this group. So I would insist to maintain the group and clarify as much as possible what kind of expertise is represented in the group. What eventually can be discussed is if representatives from the industry in trials that are sponsored by a pharmaceutical company should be part of the committee. But this is a question mark that I would like to discuss.
Sure.
One more view on the committee, or the expert opinion. Nowhere currently represented as views from patients. That might be considered a relevant component of designing a confirmatory trial.
Yeah, no, I agree with that.
Yeah. I would agree with that too. And actually, this is on the webinar, and it's getting a lot of comments already from the various followers. And one of them, actually, a particular shout out here to ALS advocacy topics, because that person has been commenting on every single rationale through the whole meeting, and so should be congratulated for this great report.
Yeah, and that's a great point. And I work with Neuronext, and we have patient advocates involved in the protocol development there, and it's worked really great. Brings an interesting perspective, both in terms of scientific input, but also kind of keeping things practical. If there's too many assessments or visits get to be too long, and trying to narrowing it down to make it a doable protocol.
Okay, well, I think we've finished with that. Thank you very much. We're now moving on to the last topic in 4A, are there special considerations for cell and gene therapies?
Okay, thank you very much. Does this advance? Here we go. I'm Shafullah from Barrow Neurologic Institute. I worked with Bob Miller and George Manasoukas on this question. When I first got this, that I was assigned to this question, I was really excited because are there special considerations for cell and gene therapies? I thought, well, this is easy. The answer is yes. And then I realized I had to kind of delve into this a little bit further. So I'm a little surprised that this is really the only question in the entire meeting that's really specifically directed at this as a topic that probably will become bigger and bigger over the next several years. We haven't devoted too much attention to this. So the possibility of gene therapy and localized stem cell therapies is very real. This is mostly owing to bioengineering technology and allowing this to go from science fiction to a reality. In terms of our strategies, gene therapy generally, actually both strategies generally aim to increase motor neuron survival and therefore then prolong life in patients. With gene therapy, this is done by essentially manipulating the transcription of genes or the expression of genes on a DNA or RNA level. So in terms of the goals, we use that mechanism to essentially silence genes or enhance genes that we think cause the disease. Or we may try to manipulate genetic modifiers
that may influence the causative genes. This is including trophic factors or RNA binding proteins, etc. The bioengineering technology I mentioned, a lot of this has to do with antisense oligonucleotides, which is really a burgeoning field, but also the vector-associated delivery of DNA and RNA molecules are the two main approaches we're using for gene therapy. In terms of stem cell therapies, we're really probably nowhere near the point where we can replace motor neurons or regrow motor neurons. And so the strategy there is to use cell-based therapies to secrete trophic factors or really enhance the environment to promote survival of motor neurons. We've got preclinical studies which really are encouraging and provide enough rationale to move forward with human therapies in a lot of cases. And in fact, we have now some data from in human trials as well. So, as has been mentioned in this meeting before, the SOD1 antisense oligonucleotide trial that Tim Miller did provided us with a lot of insights into how to design these trials. The vector delivery of VEGF that Bob Miller did was already performed and gave us information. And then Eva Feldman and Jonathan Glass's trial injecting the stem cells into spinal cord has been instrumental as well. So in terms of our rationales and recommendations, we have 6 of them. The first rationale really has to do with the fact that these are fairly novel types of strategies. They don't sort of abide by the normal laws of pharmacokinetics and pharmacodynamics. They don't wash out so easily. In addition, we've all had patients who are asking about these kinds of trials, and there's definitely a sort of a media frenzy about them, which creates some ethical concerns in terms of equipoise in recruitment. Because of this, our recommendation number 1 is that clinical trialists designing stem cell and gene therapy trials should list in the protocol the specific precautions so that we can ensure patient safety and ethical recruitment. Rationale number 2, promising results relative to safety and benefit from gene and stem cell animal studies can result from factors unrelated to the biological effect, for instance, publication bias. And so it's important that in other studies we've used confirmation in the second laboratory to really enhance our confidence in certain results for experiments. So our recommendation too is that clinical trialists designing these studies should really replicate these findings in multiple labs using both large and small animal models to enhance our confidence that these findings are real and therefore justify human trials. Rationale number 3, there are a lot of variables in these diseases: vector type, delivery method, even the immunosuppression protocol that you might use for cell therapies, the different doses, the different gene sizes. All of these things create a lot of different permutations when you're trying to optimize dose and delivery and safety. I don't think that this is really something you can prescribe in terms of how you find the optimal therapy, but I think it's important, and recommendation number 3 states this, that clinical trialists should specify the immunosuppression protocol, the vector and stem cell delivery route, the dose, the cell type, and the gene size, and the rationale for those choices in their protocol. So at least if we're rigorous about listing these things as we move forward, we can have that data to look back on and modify for future trials. Rationale number 4, and I think this is one of the most important ones, These therapies, gene therapies and cell therapies, as I mentioned, don't just wash out of your body most of the time. So there are permanent or long-lasting changes that can occur in hosts, and tools to monitor the effect and the toxicity and the survival of the cell-based therapies are necessary and require further development. So our recommendation number 4 is that clinical trialists should develop a long-term monitoring plan to assess carcinogenesis, immune responses to the vectors, to the proteins, and also the survival of the engrafted cells and other risks from adjacent genes that are activated by gene therapy. I think we probably don't know all the tools that we need to do this, so that might be included in this recommendation, is not just a monitoring plan, but also the tools to monitor this. Rationale number 5, gene discovery is still in a fairly early phase in ALS, but we have quite a few genes that we know of that are good targets for gene therapy and actually are already in development for targets. And so our recommendation number 5 is that clinical trialists of gene therapy should study homogeneous populations such as C9 or RF72 or SOD1 mutations, at least at first. Rationale number 6, this goes back to biomarkers. I know we've discussed this a lot. They're obviously needed in ALS trials, and I think with gene therapy and cell-based therapies, this is no different, and maybe it's even more important to know what these therapies are truly affecting on a biological level. And so recommendation number 6 is clinical trialists should include at least one biomarker to monitor target engagement, for example, SOD1 protein levels or RAN peptide levels. So I think there's a lot of room, I think, here for more specifics, but these are deliberately right now fairly non-specific, mostly because I think a lot of the hurdles that we're going to find are not yet identified. We're going to learn from these trials as they go on, and we're going to find more and more issues that have to be dealt with. So we wanted to make these recommendations fluid enough to be able to adapt to that. Happy to hear discussion and take any questions.
Well, it's a very topical area, and I'm sure we'll have a lot of discussions.
Sure, that was a great summary. I just have one suggestion for recommendation 5, because you're discussing gene therapy as if it's one thing. It's such as antisense oligonucleotides aimed at knocking down something, but there's a whole host of viral, vector-based gene therapies that could be much more general. And so I think the recommendation might benefit by being discussed a little bit more in general, rather than saying that all gene therapy has to be against a specific target. It can be a supplementation gene, for example.
Okay.
I wonder whether you had discussed a more specific recommendation in 4, that autopsy should be mandatory in cell-based trials. I mean, it's very strong to say mandatory, but I mean, there's a lot of reasons for wanting to know exactly what's going on in the tissues, really.
Yeah, I think that's a good point. We do want to know. I mean, that's probably the most important thing in cell-based therapies at this point, is the toxicity. How you go about that, I think, is hard because of the different types of cell-based therapy. So I'm not sure how you specify that, but...
Well, I mean, I think you specify that one of the monitoring measures should be that patients subjected to cell therapy should consent to autopsy. I mean, I think that's a quite challenging thing, but I mean, I say it because the rate of autopsy varies hugely, and there are some countries where it simply doesn't happen at all, and those are countries in which trials might be done, and they'll be therefore done to a lower standard, I would say.
It's a great suggestion.
Okay. Jean-Pierre Julien, Laval University in Canada. I noted that there's no recommendation about the placebo there. Will we inject empty DNA or empty vector into patient and so on? So maybe there should be something about that.
Yeah, I think, again, we kind of deliberately avoided that because there's so many different methods of these gene and cell-based therapies that placebo may be appropriate for some of them, but it also may be the risk of that may be high in others. So I think we'll have to think about that and how we can include that concept without being too prescriptive about it.
Going back to your recommendation number 2, I think it's quite unrealistic to say that the different centers have to prove the same evidence, have the same evidence. At the same time, what do you mean with large and small animals? I think you must be more clarify what you need.
So I don't think that the same center has to prove it. I mean, I think this can be proved among centers in multiple laboratories. In terms of large and small animals, I mean, I guess we were considering rodent models and perhaps primate models, things like that. That may be too strong.
I think we must be more specific about this.
Okay.
Yep. Yeah, Nicholas Maragakis, Johns Hopkins. I would really echo that. I'm not sure what those, to be so dogmatic that these be done in separate laboratories and large and small models. There are a lot of nuances to what those mean, and I'm not sure what those large animal models, for example, would necessarily be. So also I would echo the fact that we should really be thoughtful about hype to stem cells, and so I fully agree with that. But the idea that we should be so dogmatic about this be tested in multiple laboratories, multiple different models, that may be going too far.
Okay.
Very nice job. I did want to comment that just sort of the overall perspective is very much a safety perspective without really commenting at all on potential opportunities to do trials in different ways, look for efficacy at different time points, things like that. So I think you might think of that. Recommendation 1, it's hard to argue with that, but when is that not true? I mean, it's kind of part of the requirements, both in the protocol, the consent, the investigator brochure. So yes, of course, but it's always true. It's part of federal regulations. Then I did want to just second the comment about obtaining postmortem tissue. It's actually part of the RAC, Recombinant DNA Advisory Committee, guidance requirements to sites that they have to at least approach patients for autopsy. I think it's very difficult to require that for study participation, but at least they agree to have that discussion. And then the last one on the homogeneous patient populations, I think really more the main point is match to mechanism versus homogeneity per se. You could have a lot of variability in clinical course like in SOD1 with a homogeneous mechanism.
So that was very good. The comment I'll have, I'm not sure it's more of a comment whether you add this, but we all know this gets back to the issue of hype. We all know the enormous hype that our patients see around this technology, as exciting as it is for us. And it's an issue around consent and appropriate, really informed consent. Now, every trial has informed consent, but I would point to really what John Glass has done in the last few years, being very careful about patient selection. Now, I don't know how you put that in your recommendations, but I think we have to be cognizant that we have to be very sensitive about discussions with patients about over enthusiasm beyond the norms for most clinical trials and how to appropriately choose patients.
Yeah, I totally agree. And I think it goes beyond just the consent. It's also how do we promote these trials? What sort of advertising gets done about these trials? I mean, all of that really has to be taken into account. It's, I guess, difficult to put in guidelines, but that's something we can certainly work on.
There's published literature on the element or the issue of axonal degradation in the use of viral delivery, retrograde delivery of gene therapies, and I think that relates to the animal model pretty well in that we have good disease model, fair disease models in rodents which have a symmetry in their size and shape that doesn't necessarily apply to humans or even primates. Unfortunately, we don't have a good, in my understanding, a good animal model with that kind of geometry and the axonal degradation that would need to be tested in a new viral delivery drug. So I was wondering if there was any discussion about that, or if that, at least acknowledgement, belongs in the guidelines.
Yeah, I mean, that's something we can certainly discuss further. I think hopefully the suggestion to obtain autopsy tissue helps to answer some of that, and maybe maybe on a bigger picture, that's a lot of these studies are going to generate more questions for these guidelines than answers. And I think we're going to learn a lot in the next 5-10 years about things we haven't considered here. And that's a good example of one.
Very nicely, very nicely done. This is tough. I think one of the challenges that we're going to have in putting together these recommendations and in sort of having structured things this way is that one of the recommendations might be things are really different for cell and gene therapies under certain circumstances, and therefore some of the recommendations we've made before don't apply. But I don't know how to say that. I just wondered if there were any other sort of thoughts, and maybe we just said maybe that's the recommendation. Although I don't know that I think that that's the right way to state it. But for people who are doing cell and gene therapy, I know that you've thought deeply about how your programs are different than small molecule programs, which is sort of the aim of the bulk of our recommendations. If anyone has thoughts about how to navigate that tricky water of saying, you know, gee, some of this is actually not patently not right for gene therapy or cell therapy, not the approach we're going to take, please let us know either now or just sort of before the breakout sessions.
You also mentioned tools for long-term monitoring. I mean, for the non-comprehensive, what are you talking about?
Well, I mean, I think I don't know. I don't have much expertise in cell or gene therapy, so that's something that certainly we'd love feedback from those in the audience who are doing this kind of work. From a safety standpoint, it's going to be really important that we develop these tools. So if there's already tools out there that are useful, then we'd love to know about them, and if there are suggestions, certainly we want feedback.
The thing is, I might say that I think being general is appropriate. So tools for gene therapy might be, well, the product. So if you're making more SOD1, can you monitor the production? If you're making something that's affecting C-RANT, can you monitor that production? For a cell, obviously, could you track it? Now, I know there's technology being developed by various universities to track cells, but it would be nice, because that's pharmacokinetics. Can you track the cell? Because you call them cell therapy, but it's another drug. It's just a big bag of biological molecules. You would like a way to track that, I think. So I don't know if you need additional language or just keep it general, but the principle's there.
Tibu Siddiq, Northwestern. So yes, there would be some differences in terms of traditional looking at biochemistry. There may be a need to look at the genome in some fashion, cells that have been shedding out from the central nervous system and the spinal fluid. So there may be a need for looking at the spinal fluid or imaging studies to look at off-target effects of viral delivery, especially if the genome is being interrogated with correction and otherwise. We have seen this in mice, that you can have a lot of off-target effects, and those would have to require some sort of more detailed analysis than is done with the conventional biochemical, looking at kidney and liver function and so forth.
Okay, well, I think that's a very helpful last
Just one question. I would just like to ask a question to the audience, especially to those of us that have participated in stem cell or gene preclinical or early clinical studies. Do you feel comfortable moving to a clinical trial in human when there is a study in only one animal model and only by one lab? Or because there was a question about whether this should be done in multiple labs or whatever. What if there is a result only from one lab on an SOD1 G93A mouse? Should we move into a clinical trial, or do we need confirmation by a separate lab of the same technique, of the same vector and everything otherwise?
That's a comment from Jonathan.
Just to answer that one, just first, I'm not sure the G93A mouse is the right model. Just to answer the question about what we're doing, the protocol says that we should be following these patients to death, and that's our plan if we can afford it. That's the other issue. These people may stay alive for a long period of time, and we do continue to follow them clinically, but there are MR scans and things like that that need to be done. We do encourage everybody to have an autopsy, and so far we've been pretty good about getting autopsies, and we're publishing our data. So I think those recommendations are good recommendations, and we're trying to follow them.
TV Sadiq, Northwestern. So I disagree with this because the G93 model is correct model for what it is for, which is to show that you can knock down the SOD in a way that relieves the disease symptoms. You can make a model to show that you can do targeting of the gene within the mice endogenous SOD, but that will not relieve the disease because no disease is going to be produced with a single gene effect. So both are necessary to show that you can actually target the genome and you can also relieve the disease. So the G93 model is crucial. You can have a genetic approach, obviously, to correct any defect in a certain gene by knocking it out. But in this case, you would need both. So I think that's an important consideration.
Thanks, very helpful discussion. Thanks very much. We're now moving on to 4B, and we're going to start with an absolute cracker of the topic. What lessons can we learn from previous experience of negative phase 3 trials that were based on positive phase 2 data?
Thank you. I'm Angela Genge. I'm from Montreal, Canada, from McGill, and we have split our questions into 3. 4B is actually a France-Canada alliance. We let a few Americans in just to fill out the numbers, and we desperately thank Vincenzo for being the sacrificial lamb to this process, because before he presented and we heard from Sheriff Gary and Deputy Sheriff Bob Bowser, we had no idea how to word these slides. We really appreciate everything that has been done. We've written our slides and rewritten our slides and rewritten our slides, so nothing in the documents is accurate. You'll have to read the slides. And we would offer our country of Canada as for all refugees. November 8th, the border will still be open. And I know Jean-Pierre and I have spots at our universities, so send your resumes now. With that, Philippe, you're on.
Thank you. My name is Philippe Corsia from Tours, France, the other side of the ocean. The majority of what I have to talk about now is already talked and already comment yesterday and this morning. So it will be easier for me if it's not that good. Okay. So the background, riluzole is currently the only available drug effective in ALS. And over the last 20 years, clinicians faced a paradox, promising results in animal models in phase 2 trials, and failure during the phase 3 trials. We need to reconsider the design of clinical trials, maybe. And after a comprehensive review of the previous phase 2 and 3 trials, we identified some potential problems with the design of the studies concerning the target, engagement, and CNS penetration, and the interpretation of the results. The first rationale, did the target penetrate the CNS, and did the target engage the target? Several clinical trials focus on clinical outcome only. We have inadequate evaluation of potential biological effects of the tested drug, and negative results may reflect lack of target engagement. Our recommendation, investigators should include measure of target engagement by the therapeutic molecule and evidence of CNS penetration if not previously established. Rationale 2, important concern related to the previous trial. The design of phase 2 and 3 trial were driven more by the cost and recruitment consideration than by science, and double-blind placebo-controlled trial using drug association with riluzole, as it was the case with zaliprode, pentoxifylline, minocycline, for example, missed potential efficacy of drug under development. And our recommendations are the following. Trials should encourage trials which promote comparison to riluzole efficacy rather than add-on therapy. And the last recommendation, trials should encourage trial with 3 arms, one with placebo group, the other with riluzole, and the third with the tested drug. Rationale 3, concerning the outcome measures. The gold standards are survival and function. To definitely prove that combination of function used in phase 2 and 3 and survival in phase 3 are convenient primary outcome measures. And the second point, do the lack of efficacy is related to the lack of activity of specific biologic target? We recommend that clinical trials should include biomarkers both on disease and target engagement, probably CSF biomarkers. And the fourth rationale concerned the heterogeneity of the phenotype. This is usually not ever considered in previous trials, and it's difficult to determine a homogeneous subgroup of ALS patients. No trial promoting specific management, and it will be interesting to determine the best ALS group for a given therapeutic. Our recommendation is the following: clinical trials should take into account phenotypic and heterogeneity. Clinical trials should integrate biomarkers within criteria of inclusion, and clinical trials should consider therapeutic mechanism and patient subgroups. Thank you for your attention.
Okay, so time for questions.
Go back to your the comment about the add-on. I'm not sure I understand why your group is recommending that we not use the add-on design to riluzole. Could you explain that a little bit more?
Yes. For the example of zaliprudine, it's maybe the more illustrative example. In the patient treated by both zaliprudine and riluzole, we don't have any effect of the molecule. Conversely to the group only treated by zaliprudine, in which group we found an efficacy of respiratory function. And we might consider that the combination might cancel the effect of the tested group, that can lead to consider that the trial is ineffective, because the combination of the drug will erase the efficacy of the drug in a testing situation. No?
Well, can you go back to your exact wording? I'm not sure that we want to make add-on trials not a possibility. I think, again, it depends on the drug and the preclinical data. But ultimately, we'd like to have combinations of treatments and actually have drugs that can add on to riluzole effectively.
Yes, but it may be one of the problem of the lack of efficacy of many of the studies we have seen during the last 20 years. It's a proposition only, but yeah, of course.
So my comment has to do with, it kind of gets to the wording of the question that you had, which is, I can't remember exactly how it was worded, that failed phase 3 following positive results in phase 2.
Yeah.
And I think it's partly the question. The phrase positive phase 2 data is a little vague in a lot of aspects. And I think given the discussion we had earlier, perhaps one of your recommendations may, and maybe some for your group to consider, is maybe providing more clarity in what positive phase 2 data is. I mean, we had a long discussion on those 3 things, and how would you decide what's positive? Because a lot of it is subjective in a lot of sense. Do you have positive data?
Yes.
And I say that because I'm a little concerned that we're going too far off the edge, and we're trying to fix a problem of not having good drugs by tweaking the design. And as I said earlier, and I think you can muddle with the design all you want to, but if the drugs don't work, you're not going to solve a problem with any kind of fancy design. And part of the question here, I think, with failed phase 3 is not there may be some design aspects of phase 3, but I'm not sure a lot of it is the phase 3 design. It's having better phase 1 and phase 2 studies and more knowledge of, chance of a positive effect before you go into phase 3.
For me, in my mind, I understand this question as follows. You have a lot of study with phase 2 who prove efficacy of a promising effect of drugs. And when you go to the phase 3, you have only a lack of efficacy or failure of efficacy. What can explain this? And for me, maybe 2 main points may be advanced for this. First, the heterogeneity of the ALS inclusion patients and the design. The combination of drug and riluzole may be one of the most point that lead to lose the efficacy of the new drugs in ALS. And maybe we have to take into account what currently is proposed in multiple sclerosis, as for the example of the natalizumab compared to placebo group in multiple sclerosis. Because when you have the study comparing natalizumab plus interferon versus interferon, you have in the combined group some side effects that lead to stop the improvement in the localization of the drug because of the progressive multifocal leukoencephalopathy you have when you combine natalizumab with interferon. And we do not have these side effects only on natalizumab treatment. And I take the parallel with this for this discussion.
I mean, I agree with what you're saying. I guess maybe this is a little naivety because I don't know the literature here as well as all of you do. But are there many examples where you've shown positive efficacy in phase 2 and then not seen it in phase 3? I mean, by shown, because I would argue if you showed efficacy in phase 2, you probably shouldn't even be doing the phase 3 trial.
I don't consider it chance. No, it's not chance for me.
Yeah. So just for those of you who don't remember the zalaprodin study, there were 2 large studies performed in tandem, one with patients on riluzole and one without. The one with riluzole, without riluzole showed a slight benefit on pulmonary function. The one with riluzole did not show an additive effect. And so one possibility is, just as you state, the presence of riluzole did mask the effect of zalaprodin. The other, I think, equally probable likelihood is that this is the reason why the FDA in general requires 2 phase 3 trials, and that in fact there really was no effect of zalaprodin, and the second trial confirmed that. In either case, the masking of an effect, if it was there, was a very small effect, and I really have reservations about the idea that in a uniformly fatal disease with a known drug that has an effect, which has been called marginal here, but I think is more than that, one has to have a much more stronger justification to not use it than you presented so far. And then just as a final, I'm not aware that there is independent data that minocycline was efficacious without riluzole. And you mentioned that was on your slide, and equally not aware that pentoxifillin showed efficacy without riluzole.
Okay. Osama Khan from Japan. I think one of the failure of is come from the including the Freyram or Frey-Legs syndrome. According to the previous paper, the England group reported that if we see the ALS carefully, there are 20% of the Freyram and Frey-Leg included. So we have to exclude it as such a patient because such a patient survival is much longer than the classical ALS or bulbar type of ALS and more than 50 years old. So that is one of the problems. I think that
it's what I think. So it's maybe difficult to consider that Freyram syndrome and bulbar ALS, for example, represent the same ALS pathology. Maybe if you allow me to be a little provocative, could we consider ALS as cancer? And do you imagine to treat lung cancer as a kidney cancer? So if you make the parallel with ALS, could you consider to treat Freyram syndrome as a bulbar ALS? And maybe we have to be more stringent about the criteria of inclusion and focus on a specific phenotype, if possible, and if a dream. It's a dream, I know.
Thank you.
Just brief comment. I think in just discussing disease heterogeneity, we have to think about disease stage, early or late, because our trial with ultra high dose methylcobalamin failed as phase 2-3, but almost 40% of the group with early entry showed a very significant prolongation of survival. And so that's not exactly phase 2, but that was expected to be phase 2-3 for approval, but that failed. So we have to do a phase 3 again with placebos. So I do think disease staging it would be a kind of term that you might want in.
Okay, thanks.
Well, I think it's important that what Jeremy said is emphasized, and I think that's also what Merit was actually referring to, because if the real world is that it will be an add-on for patients, and riluzole, I realize riluzole is not the ideal drug, but it decreases the relative risk of dying at year 1 with 33%.
Yeah.
So it's not a small effect, it's actually a surprisingly large effect. But the reason why we minimalize the effect of riluzole is because we tend to draw lines through the Kaplan-Meier curves. And then we compare the placebo with riluzole. But a Kaplan-Meier curve actually is not meant to do that. And also what we should take into consideration is the curve that one obtains in the studies of Vincent and others in the first 6 months, which is the most certain part of the statistics, because thereafter patients start to die, and so the degree of uncertainty about these curves increases. If one takes only into account the most certain part of the curves, then the effect is quite large. It means 12 months in a disease which lasted only 28 months or something like that. So I would like to second what Merit and Jeremy said and repeat that what we are looking for, at least in my mind, is an add-on drug, because we should not tell our patients that riluzole is so marginal, we should not even take it into account when we are developing new therapies.
Yeah, yes. But don't you consider that the zaliprodone trial may give some strength to the proposition to avoid the combination of riluzole with
I understand. Yeah, I understand your point. So we do know that zaliprodone had an effect, and you might say, I'm going to take this further and do medicinal chemistry and try to improve its effect. But having said that, for patients, it's quite clear that marketing zaliprodone would have been useless, because adding zaliprodone to riluzole increases toxicity, because there is also a matter of toxicity in the curve, as you remember.
Yeah.
It doesn't add anything for patients to take both drugs. But I understand from a drug discovery point of view, it might be of interest to know that it had an effect on its own. But, phase 3 trials are meant to see whether drugs are worthwhile bringing to patients.
Yeah.
But I realize it's not a black and white thing.
Okay, thanks.
From the viewpoint of heterogeneity of patients and also the successful results from the lesson of edaravone in Japan, probably maybe sometimes phase 3 trials, data of that will be a starting point to analyze the positive group, subgroup, to condense or enrich the positive group of patients. Probably that kind of a positive thinking of negative results of phase 3 will be necessary sometimes. And how do you think about that?
So,
because so far, phase 3 is an endpoint of a clinical trial, but probably it's sometimes a starting point.
Yeah, it's a good question, but I don't think about this before, and I'm unable to reply with a good value of this. Maybe it's a good point we have to reanalyze, of course.
Thank you for presenting these concepts because they're very helpful. In my question, we're going to come up against comparative efficacy trials as a potential issue. And is a comparative efficacy trial a phase 2 trial or a phase 3 trial? And we should be thinking about this as part of the guidelines as well. This issue of doing something like we did with zaloprodin, the question might be, you know, if you're going to treat with riluzole, how long do you have to treat before you have to consider some other treatment? I mean, in cancer, they have that fairly well worked out. The problem is, what is the potential benefit of adding something late? I think we have to parcel out this issue of placebo control versus comparative efficacy.
Thank you.
Yeah, I have just one observation about the phase 2, phase 3 dynamic. I think it's worthwhile to go revisit the premise of the coenzyme Q10 study that was done as a phase 2. As I recall that study, it did what a phase 2 trial was asked to do. It was safe and effective and tolerable. And as I recall, the product that was given to patients had, even though it was a nutraceutical, it was certified as being biologically available. But it was not advanced to phase 3 trial because it didn't have a very impossibly high bar for potential efficacy. As I remember, a 30% reduction in disease progression. And so, you know, I think there's a methodologic comment that could be, given to this discussion on the basis of that trial.
Well, I think that's very important points for discussion. Thank you.
Thank you.
We'll move on to the next topic, pragmatic versus regulatory trials.
Yep. So we're moving on to this, and then pragmatic versus regulatory trials last. We do what Angela tells us to do. So with this heading, we actually chose to broaden the question a bit and to use this as an opportunity to kind of address the bigger issue of how do investigators interact with people living with ALS who make an independent choice to use unproven or unapproved interventions. How can we partner with them in this kind of novel and complex circumstance? And in terms of the background, many of you are very familiar with the background. Ultimately, the point is that last point, that once an intervention is reported to be possibly beneficial in a condition as devastating as ALS, many people living with ALS are eager to access it outside of the clinical trial arena. How do we address this situation? Sorry. So this is the most fundamental point is that there are some recognized, established mechanisms to make an effort to accelerate access, particularly if there are things that, when there are things, that prove to have added benefit in ALS. Now there are some active verbs missing here, but I think I would suggest that investigators should, working with the FDA, identify ways to minimize time between
completion of a successful trial and approval of effective interventions. I don't fully understand the EAP process myself, but to the extent that it may be relevant in certain circumstances, we certainly have that as a process that can be utilized. And finally, I think trialists should encourage use of open label extension in phase 3 clinical trials for several reasons. I recognize there are cost considerations here in many circumstances. Rationale 2 gets to the concern that in some circumstances in which there is a readily available intervention used in a controlled clinical trial, some subjects may obtain access outside of the course of the clinical trial, making their data, the word here is unusable, but perhaps worse, misleading in the analysis of the trial. So certainly, once again, we should encourage people living with ALS who are using unregulated, unapproved therapies to outcomes monitoring by their clinicians. This actually is references to recommendation 4, not recommendation 1. We move them around. Trialists should be aware of this possibility and assess when possible for self-prescribing within study populations. Third, there's this issue of how do we integrate data from individuals who use unproven therapies independently. Does that provide us information that we're not currently taking advantage of and accessing? And would that information be beneficial? And I think this becomes more and more of an issue, particularly as ideas are rapidly disseminated through patient forums or fora. I'm still not sure. So recommendation 3 is first, trialists should identify ways to critically assess the research value of data from truly patient-led trials to determine if there is information of utility when such trials occur. I should add that patient-reported outcomes are a different issue, but they are often included in these forums. And second, the ALS investigator community should leverage longitudinal phenotyping and natural history studies to identify or capture the use of unregulated or unproven therapies. It is possible that this could identify signals of efficacy or lack of efficacy or adverse events related to the use of these treatments or these interventions that could then either be important to disseminate or act as hypothesis generation. And finally, does the use of unregulated, unproven, or unapproved therapies raise questions about the process for trial design? And in particular, the very kind of difficult question of individuals very enthusiastic about participation, but who are excluded from clinical trials because of exclusion criteria. So we would suggest that the investigator community investigate opportunities for people living with ALS who do not fulfill inclusion criteria to participate in selected trials as a separate group, if deemed acceptably safe to do so, and of course if cost considerations can be addressed. And that phrase gets to the last caveat, which is that when resources are limited, we certainly do not want to compromise the ability to power a primary study adequately. But the question is, in certain circumstances, might separate resources be identified to address this concern that's often raised by the patient community? We would add that the investigators should design appropriate tools for this circumstance, specifically regarding randomization. For example, should such individuals be randomized, even though it's outside the primary study? tools for assessing the outcomes in this context, safety monitoring, and statistical methods. Thank you.
Okay, brought up some very controversial and interesting points of discussion there.
So great talk. I'm not sure if you and I have met, but I spoke yesterday about expanded access, and I'd love to get involved if you'd like on the writing of some of the language about these selected trials for patients who don't meet the inclusion criteria of the commercially sponsored controlled trials. So back to recommendation number 3, if you don't mind going back. I'm not sure if the words right to try ended up in the actual recommendation. Oh, they don't, just in the rationale. Okay, that's great. I just wanted to make sure that was clear. I wholeheartedly agree on the need to collect data from any kind of crowdsourced, community-led initiative. I think the Patients Like Me work is doing well, and there's an emerging industry of patient-reported outcomes providers and service providers for that. So I guess my comment, I was glad to see Right to Try did not make it into the recommendation because it's not actually a channel for access. That's a state-level agreement to turn the other way if a physician agrees to try to get a drug. There's only one case, by the way, that I believe that's occurred. There's rumors of this happening in Texas. It's unimaginable to me that any drug company in New Jersey would cross state lines with an unapproved IP, an investigational product, outside of an IND. So this has got to be something like made in someone's garage. They form a DIY drug company and then send it to people in the community. So I'm glad that, I'll just say I'm glad it's only in the rationale. But I'm happy to help with recommendation 4.
Yeah, thank you. I found your comment yesterday very valuable in that regard, as a caution that we not conflate these two very different circumstances. This is an iterative process, so we're happy to iterate with all the people in the room who know more about it than we do. The other point that we did not address, again, is patient-reported outcomes, of course, are a established now component of investigator and sponsor-initiated trials, and they can be used in either context, but should not be confused with patient-led trials.
Thank you very much, Deepu Siddiq from Northwestern. I think this is it's a fundamental issue here in terms of tension between our roles as physicians in terms of compassionate use, care of the patient, and providing the best possible and every possible resource to help with this terrible disease. But on the other hand, we also wear the hat of hard-nosed, scientifically driven hypotheses, treatments, and trials. The resources for these trials are limited, but we have seen them squandered. And you see, you have to decide whether you want it for yourself or you want it for your children. That's basically what the issue is, that we have patients who may be wealthy and want to drive the entire field and entire communities and academics to cure them or cure a family member, and we have seen that. Resources diverted and used for that purpose. I think we have to be hard-nosed about this, that do we want to cure the next generation, or do we want to waste funds and money which are limited running after wild goose chase from a strictly scientific clinical trial point of view, not from a physician with compassion and care. We need to separate those two. They fall in different categories of care.
I think our group agrees, but I think one of the issues here is, given that information is being compiled and that things are being tried, are there statistically valid ways to use that information to generate hypotheses? And I don't know the answer to that.
So David, I just wanted to say, I just figured that out. I'm sorry about that. I just wanted to say this was my favorite of all the presentations that I've seen in the past 2 days. And, you know, I deal with this all the time with ALS Untangled, and I've heard a lot of people lament the tragedy of all the self-experimentation that goes on amongst patients with ALS. I don't think it's tragic at all. I think most reasonable people, when faced with something as terrible as ALS and the fact that we have so little to offer, would self-experiment. I think the tragedy has been that we have not partnered with them
to try to find a way to track what they are doing and make some sense out of it. So I'm so happy to see that we're making some strides in that direction.
Thank you.
I did not understand how you address the issue of pragmatic trials, because this is an important issue. To me, is a good complement to regulatory trials. I don't want to say that they should replace regulatory trials, but having a confirmation of effectiveness in real life is very important. So we should address this in the guidelines and provide some recommendation on the use of pragmatic trials.
I think there's the order on this, so that's coming up.
So thank you for introducing Angela. Back. This is forward.
Oh, this is tricky. Absolutely tricky. Okay. So I am the final speaker for our group. I have to, before I even start, acknowledge my tremendous editorial staff, John Glass, Terry Heineman Patterson, and Malcolm Proudfoot, and our resource committee, David Schoenfeld and Catherine. We had a great group. We worked very hard at the 4B table all day yesterday and today, and I feel like I'm just commenting on what has been discussed for the last 2 days with just a couple caveats. So if you're really hungry, we don't have to say a lot. If you're not very hungry, you can ask all the questions you want. I will try the Leonard technique of reading off my slides, and you will comment at the end. And I do I do it in English, and I did not sense orange in the room today, Leonard. I'm just saying. Background one. So I wasn't given a real question. They said pragmatic versus regulatory. Angela, you do that. So I'm going to comment from, just read from my slides and make some comments. For me, pragmatic means off-label use of drugs that are approved for other disorders, and regulatory trials are those conducted currently in ALS in the form of the current phase 3 cytokinetics trial as good examples. I think we would all agree that for the past 30 years, both pragmatic and regulatory trials have been performed on ALS patients. And rather than listing all the references, I said just PubMed Ben Brooks from 1990 to the present, and you'll get a good overview of virtually everything that's been done. Ben, you're one of our best librarians. Pragmatic trials, yes. Pragmatic trials make assumptions that case reports and small studies of available therapies can predict benefit in ALS. And I've acknowledged Stan here, but I also should really acknowledge Steve Perrin, who, with the Gilenya question, it was the two of them that really did the work that has meant that Gilenya is not being used widely, but that was an important step in the last couple years. Regulatory trials follow the scientific method and base decisions on previous results, but do take more time, and that's a shout out to John's patient who commented on it earlier, and are more costly. For novel therapies, the regulatory path is designed specifically to protect patients from therapies that are potentially harmful, and we should never forget that that's part of what the regulations are designed to do, and to identify therapies that are effective. So the rationale one, the key question for us was, what is the role of pragmatic trials in finding effective therapies in ALS? While pragmatic trials involve therapies that are approved for other disease states, are therefore available and have robust safety data, anecdotal reports and small, inadequately powered and uncontrolled trials are not adequate to support clear efficacy. Therefore, we recommend that clinical trialists should view pragmatic trials as exploratory. Proof of efficacy ultimately requires the successful execution of an appropriate, well-designed regulatory trial. Rationale 2, although regulatory trials have led to only one approved drug in ALS in North America and Europe, so it does not, two if you count Edaravone in Japan, they've led to approved therapies in many other disease states. So trials have not failed due to the use of scientific methods through regulatory trial design. And that's a comment to say, don't throw out the baby with the bathwater, or don't shoot the messenger. Because a trial fails doesn't mean the trial was poorly defined. It may be that the therapy was ineffective. Just imagine. Continue to follow a recognized pathway of drug development from preclinical to phase 4 trials. That's a comment of David Schoenfeld, so imagine me as David. Recommendation 2, trialists should continue to use a scientific method to identify effective therapies and to improve success through improved biomarkers, therapies, and outcome measures. Trialists should include adaptive, futility, and other trial design innovations. So Lauren, my counterpart from Toronto, went over some alternative trial designs today that we should all consider. Rationale 3, what are the issues in regulatory trial design? Because there are some. Regulatory trials are dependent on strong preclinical data, sensitive outcome measures, and biomarkers that correlate with disease severity to develop ever more effective therapies for their patient population. These trials require sensitive and relevant outcome measures that are improved by adjusting to new scientific breakthroughs. And so recommendation 3 that I am putting forward is that trialists should require strong preclinical data on developing therapies. Trialists should also identify more validated biomarkers and use outcome measures that are sensitive to change. Trialists should be prepared to improve outcome measures based on new scientific information. So outcome measures that were chosen in 1990 doesn't mean that they're the same outcome measures we should choose in 2016. There has been science. We should use the science. Rationale 4, how does the ALS community take advantage of the orphan disease programs? And with it, I want to highlight something that hasn't really been discussed, but I think it's really important, and it speaks to the speed of drug development and the interest not only by the patients, but also by the regulatory authorities that we speed up drug development in orphan diseases. So both FDA and EMEA have very specific special programs that some of the current companies in the field are utilizing for orphan diseases, and the system is streamlined to encourage pre-registration meetings, new biomarkers, new outcome measures, and the carrot thrown in for companies is tax credits and the ability to obtain approval of a successful drug with one registration trial. So this speaks directly to the gentleman's comment that there are mechanisms in place, but we have to use them as efficiently as possible to not slow down the process. So clinical trialists and their partners should take full advantage of the orphan disease programs of the FDA and EMEA. Trialists should develop and present novel outcomes to both authorities and novel trial designs to regulatory agencies at the pre-IND phase in order to accelerate development of effective therapies. And with that, I'm open to questions.
Very well presented. I think Hiroshi's got I want to ask a clarification. When you say pragmatic trials, you said drugs approved for other disease. How about not approved for any disease, supplement control or something? How do you define them?
I didn't define those. I guess we would include nutraceuticals in pragmatic trials, but if a drug has no IND, if it's not approved, we don't have access to it. I'm not talking about gene therapy, but we have access to nutraceuticals and approved therapies for pragmatic trials. If the drug hasn't been approved and is therefore not available on the market, then it has to go through a regulatory process anyway. And so that's how I looked at your your question to me, pragmatic versus regulatory.
I have a different explanation for pragmatic trials.
Okay.
So this should not include only trials evaluating drugs with different indications, but they should include also trials that are usually done in clinical practice, testing drugs that have been found effective in regulatory trials, and looking at the efficiency of these treatments in real life.
Okay.
And this is something that is very important because you perfectly know that regulatory trials show some benefit in a very selected patient population. And we don't really know what happens in all those that have the same disease but were not enrolled in these trials. And in this regard, pragmatic trials can be very helpful. Obviously, they are dirty. The only experimental approach is randomization. For the rest, the patient is evaluated as in clinical practice, but otherwise the message that comes from these trials is generalizable.
So I would actually, what you just mentioned, Dr. Beghi, I would consider those phase 4 trials or post-approval trials, because you're talking about using a drug in a population for which the drug is broadly approved, not taking a drug from oncology and using it in ALS, but using a drug that's been approved for ALS or epilepsy in patients with epilepsy. So I'd call that a post-approval, not a pragmatic. For me, I made the narrow definition. I think Terry was my editor on this slide, and so she must have agreed that pragmatic is using drugs from other fields. So minocycline would apply. Gilenya would apply, these drugs that are actually approved in other fields and trying them in ALS. That was my definition I worked with.
I may agree with you. Probably there is some confusion and misunderstanding in the definition, because if you look in dictionaries, pragmatics may have also this meaning that I mentioned.
Okay.
So I'm also confused about this distinction in many ways. So it sounds like if Hiroshi's interpretation of your definition is correct, a pragmatic trial is any trial with a drug that is basically being repurposed but is available. If that's the case, then I think your rationale is fundamentally wrong, because if you look at the data that led to topiramate, celecoxib, ceftriaxone, pioglitazone, lithium, none of that came from case reports. It all came from preclinical data. And so it's the development pathway is in some ways not that much different than proprietary drugs. So I think that has to be at least considered. The other thing is that pragmatic trials can also be regulatory. So, a phase 3 trial of ceftriaxone, for example, had it been positive, could have resulted in a relabeling and a completely different indication for that drug. So again, the regulatory pathway is different in the sense that the toxicology requirements may not be as rigorous, although that was a big problem with that trial. Whether that was an issue that had to be surmounted with that trial. So I'm just not sure why we need to be going here. I mean, the use of repurposing drugs, I think the scientific method is pretty much the same for that, or for non-approved entities.
So, Jeremy, I think you're actually agreeing with one of my recommendations, which is if you have an idea of an approved drug that you want to use in ALS, you should follow the regulatory pathway. So you and I are not disagreeing. I'm using pragmatic in a particular way. Once you've made the decision that it should be tested in ALS, then you step into the regulatory pathway. But so the pre-Neil studies in topiramate, the reason you tried topiramate in the first place, was it because you woke up at 3 o'clock in the morning and said, oh, I should try topiramate? You had a reason that was based on something before you had the thought.
But it was exactly the same as would happen in proprietary drugs, which was based on animal preclinical data.
Right, so you followed that pathway.
Sorry, Angela, I guess I wanted to add to what Ettore and Jeremy have said. I understand pragmatic trials to mean something very different, and I don't know if there is an official or formal definition, but this has come up, I think, in the context of this.
We do actually have an official definition here.
There is an official definition. It's more with what Corey said, but pragmatic trial, a term first used in concert has an extension for it. That can be broadly defined as a randomized controlled trial. The purpose is to inform decisions about practice. This extension of the concert statement is intended to improve the reporting of such trials and focuses on applicability. So that's obviously important that we get that. You know, I don't know what the intent, Hiroshi, of your question was, whether it was more what Corey meant or what...
Yes, so I think that this has come up in PCORI and talk about comparative effectiveness and doing trials through electronic records and that sort of stuff. And I worry that we're sort of missing the opportunity to give some serious input and recommendations into how those sorts of studies should be done, because it's not clear to me that there really is sort of a good methodology for how that should be approached. So I worry that maybe we're missing an opportunity.
We could be. It was our interpretation of the phrase, Michael, and it didn't include PCORI's approach.
Maybe, you set it up this specific research question. Would you like to make comment? Also, just to point it out, CONSORT and also NIH has definitions for this the terminology.
To my knowledge, a pragmatic trial is a trial that is done in real practice, in which the only element that is introduced on an experimental basis is randomization. So the patient has the disease, the patient qualifies for the inclusion in the study, and then he is randomized to receive the experimental treatment or something else, and then everything is done as in usual practice. So there is no change in real practice. This should be a pragmatic trial. And compared to the regulatory trials that have very high validity, internal validity, these trials have very poor internal validity, but they have great external validity. That is, that the results, if seen positive, can be applied to the whole population of patients that have the disease in question. This is my interpretation.
But then how does that apply to ALS where we have real results? So I looked at it as pragmatic being using drugs that are not approved for ALS, and your description of pragmatic fits with my interpretation of what a post-approval trial, real-life trial is. So we were happy to rewrite again, but I think that what you're addressing is different than what I spoke to.
So I wouldn't throw out the recommendations. I would just make sure that the definitions are clear, and then you might want to have...
I might have to add another...
Right.
Okay.
What we've done is useful the way you define it.
Thank you.
Okay, well, thanks very much for... We've now got one last bit of work to do before lunch, and that is reconsideration of the guidelines. So this is guideline number 16 on page 84 of the handout, which is relevant to all of the discussions we've had in this session. So I might just start by reading it out. Investigators must always conduct clinical trials in accordance with the statements in the Declaration of Helsinki and in conformity with good clinical practice. They must fully inform patients about all aspects of their participation in the trial, ensure that patients understand the diagnosis and its implications, minimize the physical and psychological burden of participation, and avoid any discrimination in screening beyond inclusion and exclusion criteria. Open for discussion.
Agreed. Everyone agrees. Yes. So no question, as it is.
Michael wants the question.
Yeah, I just wanted to ask whether it's really relevant to include here that patients understand the diagnosis and its implications. That's obviously important. I'm just not sure it belongs in a trial guideline in the sense that people have a diagnosis before they're screened for eligibility in a trial. I guess just wonder whether this is going beyond the realm of what we need to address.
Well, I think it's a point to take on board. Maybe it was a point before its time. Bob, did you know that? You give it a thumbs up.
So this is Angela. You're taking this question? 16? Yeah. All the 16 questions. Who's taking it? Question 16.
Oh, wait a minute. You gave the question from the previous guideline?
Right.
Yeah.
Yeah, we just said yes.
Oh, yes. Yes.
Hiroshi?
So,
If I could. My interpretation of that clause was to say that patients should understand what options are available outside of the context of the trial.
Yeah.
okay, so maybe Angela, if you take those comments, and if you needed to change, you have to go through the same process. Right, same process of the verify method. Right. So I think we finished on time. So we reconvene 1 o'clock, and we're going to finish the clinical, very important final session. Thank you very much.